Well, first I want to say to our in-person and our online group, a big welcome to the first day of FlowTex. We're really excited to put this on for you. Today is our first test of doing a more. Are we on? Yeah. Oh, well, let's start all over again. So, all right, everyone. Well, welcome to FlowTex. This is first day. This is our kind of a newly adapted practical day where we're gonna go over everything from fluorescence to panel design to kind of high dimensional high parameter panel analysis, and we're really excited to bring this to y'all. I do have some various key things for our in-person groups. So first of all, we have our bathrooms. There's bathrooms located directly behind this auditorium, as well as across the way in the vendor and food area and in the UTIMM direct building. We do have a punch card for everyone wanting to participate. Go around and if you get at least half of them punched or stamped or whatever, you can turn that in at the end. We are going to be giving away a lot of prizes at the end. And then we will, and for the online people as well, we will have a couple of online raffles as well, or include the online audience at least on it as a chance. We're still working out that detail. So punch cards for some of the raffles. No food and drink in the auditorium. This is to keep it nice and clean. You can see they're very nice to let us use this facility. Let's try to keep it all pretty for them. We do validate parking for the directly adjacent UT parking complex. Just we'll have those parking validations at the registration table throughout the conference. Please remember that to visit our vendors, we are here through their gracious support. As all y'all know, we do not charge anything for the conference itself. This is all supported through vendor support. And we're very lucky to have this collaboration. And we're really excited to bring you such incredible content. I mean, I'm also the treasurer this year. And so I got the great benefit of seeing all the costs of the flights for being back in person. And the hotel and all that, I'm really kind of excited to get that bill at the end. So, but if it wasn't for our vendors, we wouldn't be able to put this on. And we'll be putting rotating up our vendor support through it, and we'll also in the closing ceremony take a time to thank them personally as well. So and let's see here. So welcome everyone. I hope everyone's doing okay. If there are any issues that you need us to help with, just come and visit us. Either find one of the committee organizers, or go up to the registration table and they'll help find it. The first talk today is going to be Kelly Lundsten, and I'm going to get her slide deck up here. All right. Okay, share it. No, I don't know. I should have been at the practice session. All right, I need to quick share this. Sorry, it's back to Zoom. I really was hoping I got rid of Zoom. Was that?
Through the clicker?
It's through the mouse. Oh, here, Sarah's going to take care of that. Well, let me while I do this. So Kelly is a flow veteran. She's a subject matter expert in fluorescence chemistry. I've known Kelly for a long time, since I believe it was either one of the first cytos that I went to, or actually it might have been before that at one of the courses. But it's always an absolute joy to work with, and it's so fun to be in the room with someone who gets so excited and geeky about fluorescence and flow cytometry. It's crazy. Originally, I remember when she was in molecular probes, and she's had a wonderful career path since then. And she's currently with Fluorofinder, really helping people do advanced panel design and a lot of other outreach and fun stuff. So we're really excited to have Kelly talking today on fluorescences. And so please.
Thank you. Oh, how's everybody feeling today? A little early, got a time change, so probably to make me go at a normal person speed. So I've been the fluorophore girl like my entire life. So it isn't that we all expect each person to be an expert in all of the elements surrounding flow cytometry. We all have our own little path. I was with molecular probes when I was really young and just became an application scientist for the vast majority of my life, supporting different kinds of high content screening, microscopy, flow assays, and that sort of stuff. So if you have any questions about any single type of probe that ever existed, we used to have to memorize the molecular probes handbook, which was about that thick. Yep, that's the reaction you should have to that. That was considered training, was just sit there with that book. So, but today we're going to talk about kind of how the chemistry, in a practical sense, I liked that introduction, a practical sense relates to flow cytometry. It isn't that you have to be an expert, but it's really important to think about what it feels like in space. Are we happy in this space? Are we not happy in this space? How is that going to impact my MFI? So I'm with Fluorofinder, and if you're not familiar with Fluorofinder, we are an online application to help people do panel building and spectra viewers and all sorts of other stuff. I also do consulting, and they were one of my consulting clients. And I'm like, this is a big project. We want to make a very, very, very intelligent panel design tool. Kind of like, maybe I'm dating myself, but I used to pour my own gels. I'm just saying, who does that now, right? I mean, I think that having a little help at getting us, just a more intelligent place to start from isn't really going to hurt anybody, as long as we understand why we're doing what we're doing and we know how to catch all of our, we know how to read our data in order to catch problems. So let's see. There has to be a way of going forward. There we go. All right, so I'm sure you've seen these sorts of things before. There's certain things I want to teach you as we go through each point. This is relatively academic. We're not affiliated with any particular manufacturer. It's just to give you the overview of all of the fluorophores that exist in the market right now. Yes, but that means I have to stay behind the computer. No, it's fine. I can stay in one place. It won't kill me. So mostly, if you think about our eyes as detectors, we have a certain quantum efficiency across the visible range. We can see in the middle of the visible range most efficiently. That's when we're at the highest quantum efficiency for detection. So green is easy for us to see. It's easy for contrast, easy for chemistry. There's a reason why most of our endogenous autofluorescence emits in the green. Green is a happy place to be. When we start getting towards the blue, which is going to require a much higher frequency energy input, like the sun, we have a lot of endogenous autofluorescence to protect us from the sun. But as it comes to be a reagent for us, that is not a very efficient wavelength of emission. It's a very small structure. It can't absorb very much energy, and therefore it's very difficult for that to be bright. And that's why any way of polymerizing fluorophores was just really, really important for us to be able to use UV and violet lasers. I was around during the time of QDOTs, and it was not the happiest time in my life. It's really nice that now we have organic light emitting diodes or different DNA backbones that we can start to make better fluorophores. And then once we get into the deep red, we have exactly the opposite problem. The structure becomes very big. It can absorb a lot of energy, but its quantum efficiency goes through the floor because it's hydrophobic and it doesn't want to be in an aqueous environment. So that's where it was really important for us to figure out little tricks about how to keep it soluble and how to keep it happy in our naturally aqueous environment. So the one thing that I want to point out first is things that we often overlook. From a molecular probes person or like a fluorophore person's perspective, the fact that we normalize spectra is really annoying because it makes us believe that all things are created equal. And we know that it's not. We know it in practical application. We experience it every day. But having these kinds of tools are just helpful for matching filters, not for giving us a lot of information about how that fluorophore is going to play in the sandbox with my other dyes. So if we look at something like PE and PE Sci5, they are both very, very bright dyes, obviously. And when they're normalized, it makes it seem like all is well in the world. But when we use a more optimal excitation for PE, obviously we all know this, we're going to see a greater output because we have a greater efficiency of excitation going in. And we have an enhanced output. The Alexa 647 in this instance wasn't excited by either 488 or 561, but it is always excited by 633, and that's why PE Psi5 is so problematic. When you think about how PE Psi5 is constructed, you have about 3 to 4 Psi5s conjugated to the PE, but that's how many dyes you have conjugated to an antibody. So to the cytometer, when it has cross-beam excitation, they look identical. If it wasn't that we had compensation and we had the ability to spatially align our lasers in order to give that time delay, the instrument wouldn't be able to see the difference between those two things. The other thing that I want to point out when you look at spectra, just to keep in the back of your mind, all of us have grown up together in flow, and there used to be this time where the debate used to be whether or not compensation hurt your data or not, right? Stuff that we don't even discuss hardly anymore. And I still hear it when I do training, when people are like, oh, PE Psi7 is killing, everybody has like a dye that they just dislike. And they'll say, well, it's over 100% compensation. And we all know that spectra is a fingerprint. The spectra can't change because it's a spatial distribution. It is a statistical distribution, sorry, not spatial, of the likelihood of a photon at a certain wavelength emitting from a fluorophore at any particular wavelength. And that statistical distribution is like a fingerprint to that fluorophore. And that's why we use other modalities in imaging, like lifetime imaging or other photophysical characteristics of the dye, that when they're in close proximity or in an environment they don't want to be in or something like that, they'll change lifetime, but the spectra will remain the same. There are ways of shifting spectra like FRET, or a lot of people aren't familiar with the term homofret. It's when two molecules of the same spectral characteristics get too close to one another and they start to resonate energy as a larger molecule. They shift red. You see it with nucleic acid stains all the time. But those are the only specific conditions where two fluorophores are close enough together to change spectra. Otherwise, when we say something like, I have 100% compensation or something like that of, let's have a good example here. BUV 661 into APC. It's a big spiller. It absolutely has excitation off the 633 laser, but there's absolutely no chance that spectrally that dye is having over 100% contribution to the intended channel for APC. Obviously, when you see things like that, it's not the dye. The dye isn't the culprit. You need to fix your voltages because you're out of balance, or don't care. That's also a possibility. It depends if it affects your resolution or not. So that's definitely a myth that we want to dispel, is that there are these kind of entirely incompatible combinations. There are some that are not as beneficial to have together in co-expression, but every single one of these has their own personality. We don't think of all Alexa fluors as being equally good or equally bad. That doesn't make any sense. It depends what the context is, and it depends what the instrumentation is. All right. This is all super, super, super basic stuff. But I love to act like I'm a fluorophore. So I have dreams about being a fluorophore. So absorption and emission. Any molecule that's capable of absorbing energy and having it reflected, obviously, is a chromafluor. But that is like, if you've ever seen the structure of phenol red, it's basically just a broken FTSE, because what is required for any sort of fluorescence is that the molecule remains rigid. It is a semiconductive circuit around each of those conjugated hydrocarbons. And if that circuit needs to remain intact. So photobleaching is when the ROS from our aqueous environment interacting with energy coming from the sun that breaks the water, creates ROS, and attacks the first thing that it can quench its thirst on, which is the double bonds of a hydrocarbon. So that is when the molecule breaks, and all of a sudden it has a little bit of a wiggle, and it is not able to ever be fluorescent again because you can't reconstruct a conjugated ring structure. If you see a fluorescence that dims or even goes dark, and then it becomes fluorescent again, which you can regularly see in microscopy, it's because there was a condition of quenching, and the quenching was released. It was never photobleached. Another thing that people used to say all the time was that with PE size 7, their size 7 would cleave off their PE. That's actually not totally true either. Size 7 is a very elongated structure. He probably just lost one of his arms through an ROS interaction, because those are a bunch of double bonds that lead to with a fluorophore. It's a mirror image of itself. So when the molecule jumps to its exciting state, so absorption and excitation, we tend to use those two things interchangeably. And they're not necessarily, there are subtle differences. Excitation is when we monitor the emission at a single wavelength, theoretically the peak of the emission of the dye. And then nanometer by nanometer, we scan the entire spectrum in order to see how many photons emit at that wavelength based on the excitation wavelength. So with excitation, that is actually the energy that's generating the photon, right? That's going from input to output. When the molecule jumps to its excited state, you can imagine that it is going to start to vibrate. It becomes hot, and it starts losing energy in motion, mostly vibrational energy. And that's called a non-radiative energy loss. That energy loss is how we get an emission different from our excitation. So it has to decay, or else it's not useful to us, because we would be detecting it at the same wavelength that we excited it at. Phosphorescence is also useful, but mostly for microplate assays or something where you want to have a time course, or but for not so much for flow. They can be fluorescent and phosphorescent in some instances, but that is very specific instances. All right, so mostly when we talk about brightness, we're talking about context. You have the single brightness of a dye, blue dyes being super dim, deep red dyes being a little bit brighter. But really what that's comprised of is the ability for it to absorb energy, or the extinction coefficient, versus the efficiency with which it generates that energy or resonates that energy until it becomes an emitted photon. That's a very fast process. That's what lifetime is. When you hear of people talking about blinking, sometimes blinking is desired or not desired depending on the microscopy application. For us, we don't even notice, because we have a very fast technology. We don't rest in a single space in order to detect its blinking. But it's basically rest to regenerate before it can actually absorb more energy. So there's the single brightness of a dye. And when we talk about bright dyes, that's usually what we're talking about. But the brightness of the antibody is what we're actually detecting, because I can only have one PE to one antibody versus 3 Alexa 647s to one antibody versus 5 FITZs or 5 Alexa 48s to one antibody. You know, you have this ability to have additive intensity. For example, this is why it's so important that we can polymerize. We want polymers that allow each monomer to be individually unaffected by its neighbor. So it just, as a unit, is constantly becoming fluorescent and coming back to its ground state, right? Independent of all of the other fluorophores in the polymer. And that is actually a really tall order. It means there needs to be a certain amount of distance between each of those monomers. But theoretically, the reason why we want to polymerize is because the longer that chain becomes, the brighter, and that's it. The spectra always remains the same. All we're ever doing is increasing the intensity by increasing the size of the molecule. And then you can have a one-to-one because you know how many of those, because you know what the brightness of that single molecule is. And all you have to do is take that and conjugate it to the antibody. But in situ is an entirely different thing. And it's a bigger deal in microscopy because we have so many different sources of endogenous autofluorescence. Tryptophan it's endogenous, right? Let alone vitamin E, vitamin C, vitamin whatever. All of your vitamins in your body are designed to scavenge reactive oxygen species. That's why we don't all have cancer. And they are all fluorophores. They're all endogenous autofluorescence. So biotin, all sorts of metabolic cofactors, any cell that's rapidly dividing is going to have more nutrients and therefore more autofluorescence, like NADH, for example. And I actually have a training once a month now with Fluorofinder. One of the reasons why I love working for them is I get to do more training. So it's one Wednesday a month where I do like a deep dive into everything that comes before this. It's not even about dyes. It's mostly about sample prep. In microscopy, we think of brightness per pixel. In flow, we think of it per event. So one thing that I really want to challenge people to think about more critically is the fact that most of our cell samples are not homogeneous. We take a biopsy or we take a heterogeneous population of cells. Maybe we have an activation or a disease state or something that's causing a portion of those cells to become more metabolically active. We now have a third degree of variability because say that we polarize some T cells. Not all those T cells are going to be polarized the same way. Not all of them are going to be activated. Some of them will still be inactive. That is now a different autofluorescent background than those that are activated, let alone what else is going on in that cell sample. And that is why controls are so important. I was working for the last year with Thermo on the Bigfoot project, and I had never done cell sorting in my entire life. I have literally been doing more than 20 color flow the vast majority of my career. And I had no respect. I've got a modicum of respect for cell sorting. Now I understand why there's dedicated operators for things. I definitely learned how the sensitivity of the instrument can impact the results, specifically using PMTs versus APDs. You can get more NIR sensitive PMTs, but they're pretty expensive. And the difference between a jet and air sorter and a cuvette sorter. Both of those things gave me a new respect for it, because you're kind of thinking of like the attrition of light. You have a single photon that's emitted. What percentage of that photon actually ever makes it to what is being represented in your data through the attrition of signal throughout the entire process? All right, so with the Stokes shift that I was talking about before, the non-radiative decay, that does not produce the photon. Initially when we started developing dyes I've developed a lot of dyes in my life. I was with Biolegend for like 11 years, and we were specifically even starting to make dyes for the Aurora. Dyes that we couldn't use on any other platform because they weren't going to be useful. But typically these dyes that we're accustomed to that have higher efficiency, they're all right in that visible range, and they've got a really tight Stokes shift of a couple, about 20 nanometers or something akin to that. That's fine. If you want to have a longer Stokes shift, as in a single molecule that's capable of being excited short, and that emitting a lot longer than the rest, too much energy will have been lost in non-radiative decay for any of them to be bright. This is why we have to have tandems. So when I talk about good dyes and bad dyes, bad dyes are usually because they assault other channels. We don't like culprits. Good dyes often tend to be the victims of that assault. So when we think of the chemistry of dyes, good dyes are relatively bright and or tunable in their brightness. They are excited by still a limited number of excitation wavelengths. We don't think that any of us are interested in always having to use 9 laser instruments. And then things that are more prescient to us are solubility, because as we polymerize, it was the same with Q dots. These things don't want to be in solution. So we have to basically pegylate them until we can force them to want to be in solution. That makes the molecules really big and often have a lot of nonspecific binding. And more importantly, discrete excitation off that, like if BD just came out with the real blue and the real yellow. I have an idea about what they are. They're actually very small for what a polymer should be. So I can kind of guess at what they are, but they're not very revealing about what they are. Surprise, surprise. I even did a patent search on it and I couldn't find a patent on it. So that's also very telling. The patent's already issued. Anyway, but one of the reasons why they're going to be so valuable is because same with the NovaFluors, because we can control the wavelengths of that core polymer, we can make them more discrete to be only excited or selectively excited by certain wavelengths and not others. And that proteins do not allow that for us. And then the other most important, there was a lot of most importance in there, is that the donor and the acceptor, in order to get that Stokes shift, are as efficient as humanly possible. Someday, I do believe that we will have chemistry that allows us to have 100% transmission of efficiency of energy between the acceptor and the donor. And that would be as if then they're one molecule. Right now, we work with two, and we have to handle the problems of both of them, including the photobleaching and the solubility and such like that. Okay, so if you look at, I don't expect you to remember any of this, my favorite slide. So as you can see on the UV end of the spectrum, the molecules are going to be really, really small, like tryptophan and NADH. As they get into the near infrared, they're going to become much, much larger because the energy out in the deep red is a lot less intense. So you need more absorbing elements of a structure, of a conductive structure, in order to be able to even be excited by that low of an energy wavelength. So you can see size 7 over there. Size 7 is extremely susceptible to photobleaching. If I were a reactive oxygen species wandering around my test tube, I would look at that and be like, lunch, because it gives me everything that I need to neutralize myself. So basically, they're just kind of like traps for ROS, and that's why they photobleach so quickly in our applications. Also because photobleaching is a perception. You can have a particular rate, but the point at which we can no longer see something, we'll say it's photobleached. Doesn't mean there's not any of it around. It just means our perception has hit a resolution limit. So on the right-hand side, this is usually how I explain what I do to people. I'm like, I study fluorescence chemistry. Let me tell you about your antioxidants. So if you look at vitamin E, that long carbon chain right there, that is, that's what makes it insoluble. And that's what helps it intercalate into the lipids of your cell surface, basically. And then you obviously just have a little fluorophore sitting there ready to do its job. Vitamin C, on the other hand, it needs to protect the inside of us from cancer, because it's not just light that's producing ROS. All sorts of processes are. So it's important that that molecule is highly soluble, which it is. You can see all of its soluble points all around. And then I always like to put riboflavin up there. The flavonoids are also highly autofluorescent, and that basically is just like fluorescein. If you look at the structure of FTSE over here, that bottom ring is actually not part of the fluorescent unit of that, of what's generating an emission from that molecule. It kind of just sits there as like a place to be conjugated to an antibody. So they are basically a very, very, very similar structure. So quenching, we've already talked about. More is never better. We can't just keep increasing brightness by increasing the number of dyes. And we use this effectively in all sorts of enzymatic assays when we actually intentionally quench a peptide or a protein by having the fluors too close to one another. You can kind of imagine that a fluorophore might be planar, but when it jumps up to its excitation state, its pi cloud is not planar, right? Its pi cloud expands. It becomes a much larger, more sensitive structure in space. And if another fluorophore is too close to it, the electrons in their orbits might collide with one another. And that's absolutely normal. It just temporarily brings it down to ground state before it's able to be excited again. We see this with DAPI all the time in mounting media. You get a black hole at the center of your nucleus if you leave it in the mounting media. So in this instance, say that this was my initial conjugate, say that it was FTSE, and I've got 4 of them. They're all spread out, they're happy, they're independently unperturbed by each other. But if I try to increase the degree of labeling, all I'm going to end up doing is quenching the neighbor until I can photobleach away one of them. If I destroy one of these 2 fluors that are quenching each other, the other will be released from the quench condition. So it's really the only time that you see an increase in intensity. You can't gain energy. You are only ever losing energy because we're in an open system. So we still rely really heavily on proteins, but GFP changed all of our lives to be able to endogenously see the expression of proteins. Their biggest strengths are their ability to be mutated into different colors off of the amino acid residues. The important thing to know, though, is that within the beta barrel is encased this tiny little chromophore. The chromophore itself is non-fluorescent unless it's in that beta barrel. The problem with proteins is that the scaffolding is entirely important, is entirely required for the fluorescence of any of the chromophores that are embedded within that protein, which means their sensitivity to solvents is going to be horrid because the minute that they become even partially denatured, the entire complex falls apart. Which is why we can't use methanol fixatives, we can't freeze them, we can't whatever them. They do allow us to quantitatively look at the expression of proteins as long as they're not affecting the health of the cell or the ability to transcribe that protein. Let's see. Oh, I remember when I was first at Molecular Probes and these were coming out. It was kind of funny because they would have a protein that would express a red, like an RFP, and they would want to track that protein, and the proteins would just be dragged along by their RFPs in order to dimerize or tetramerize. So the localization information early on was not taking into account that these molecules don't like to be by themselves. Just like PE, they want to be able to be folded into a larger tertiary structure. There's a website called FP Base that I highly recommend. It's out of Jennifer Waters' lab at Harvard. She's the flow core or the microscopy core director there. Also, a lot of these guys require cofactors, which can actually be really helpful because in live cell assays, if that cofactor isn't there, they won't come together. So that is actually a way of reporting on endogenous biological activity. The phycobilly proteins are multiple leaflets that are folded into a much larger structure. Strengths are obviously humongous extinction coefficient. But when we think of that protein is just the scaffolding. It's 240 kilodaltons, 240,000 kilodaltons. Daltons. That makes so much more sense. That would have been a that would have been a humongous molecule. So that is actually still humongous when you think that an Alexa Fluor 647 is only 800 to 1,000 Daltons. And it has to be folded into this kind of big tertiary structure. I call it Holly Hobby hair. If you've ever had a Holly Hobby doll, maybe that's also dating me. But you can only have a 1-to-1 with an antibody. If you ever did get 2 PEs on a single antibody, first of all, it wouldn't purify correctly. You'd have this monster band. I don't really recommend conjugating your own. It requires a partial denaturation of the PE in order to be able to get into, in order to be able to cross-link it through the hinge region of your antibody. And then you're hoping to refold it and recross-link it. It's not the nicest thing to do. Weaknesses are just tremendous. Mostly it's the synthesis part of it. It's really, really, really cheap, and that's great because we have enough sources of algae to purify these phycobilins. But it would be nice if we just had a synthetic version where we could control where all of the fluorophores are. I like to think of phycobilly proteins as being the original, the OG polymer. It was like nature's polymer for, because within it are these. Oh, okay. Oh, I was pointing it at myself. These guys right here, these white threads, those are theoretically the fluorophores. The rest of this is just, oh, sorry, it's not the white bits. It's these pink bits here. I'm like, that does not look like a fluorophore. The pink bits here are the phycobilin. It's the single fluorophore unit. You have like 15 of them in a single PE, but they have to be attached to different parts of this protein in order to be rigid and be fluorescent. This is the same as the quenching. You have a certain distance when we talk about fret. The fret is not between 2 antibodies or 2 proteins, it's between 2 fluorophores. So you have to know where those fluorophores are in order to say that when 2 things are bound in close proximity, that the fluorophores themselves are close enough to tell you the distance between those 2 things. So engineering, the ability to engineer things is what we've kind of not been lacking, but it'd be great if we didn't have to use nature, rely on kind of nature's limitations in order to do this. And then as I was talking about before, just the photobleaching curve of any sort of photobleaching depends on how bright it was to begin with, because the minute that you hit a point where you can't see the signal anymore, well, was everything equally bright to begin with? So when we see photobleaching curves like this, they're all normalized to 100%. They're not starting out with the difference between how bright they were to begin with. Really important to know in microscopy. Not that this is a microscopy talk. Apparently that's what I want to talk about today. All right. So this is from, how much time do I have? Oh, I have like 3 slides. All right. So this is, if everybody, does anybody know Ruud Hulstus? God, I love that guy. He loves to test the limits of our technology. And we already know that this is true. When we were developing the Brilliant Violet dyes at BioLegend, we realized that we had to conjugate BD785 in a red room. Literally, they have to do their conjugation chemistry in a room with red lights because we know how bad even residual light is for photobleaching. We moved into a new building halfway through the project, and we had to test all of the lights in every single room to figure out what light bulb was going to be best, what kind of light structure, whether it was a long, just, it was absolutely ridiculous. So we had to go through it like it was a scientific process. What we found out was that diffusers are clutch. Having any sort of ball of like bare bulb was not great. And having beautiful big windows that staved off any sort of, seasonal affective disorder was also really bad. I always like to say, if you're driving a car and your arm is next to a window and an hour or 2 into it, you realize that you are now unequal in color, that light is much more intense and much more specific for the function of the fluorophore, therefore more efficiently exciting those dyes than any sort of, even this kind of residual light. So in this instance, he was taking PE-7, looking at the kind of degradation or the photobleaching of each of these. And even under just 4 hours of continual kind of exposure at these different intensities, which we also had to do. We had to have one of those lux meters, those next to our lights in the room. That, you could see this significant difference into the spill back into the PE channel as the Si7 photobleach. And the way that kind of manifests is this was actually just under normal storage conditions. So that's the number of days that just the vial was in the fridge, was taken out to be used and applied. This is why, no matter what anybody says, you have to have the dye for the experiment used as your compensation, let alone your mixing control, where absolutely you'll notice that there's a difference in those things. You cannot go back. So what I like to kind of tell people is, because oxygen is the great destroyer, but also the great creator, the more times that I open up my vial and expose it to another influx of oxygen, the worse off my reagent will be. If I only ever opened it once, aliquoted it, and then closed them up and actually just stored them that way so that they weren't repetitively brought into the warmth, weren't repetitively left for 4 hours on my bench, were generally just not stressed out or exposed to oxygen in life, those will be a lot more stable than my normal antibody. So it depends on how quickly you go through your reagent, but minimizing any exposure to oxygen is the way to go. Okay. So I'm not a big proponent of Q-dot nanocrystals, but I do want to talk about them because we're getting into a more advanced technology called graphene particles, or C-dots, carbon dots. And we need to know kind of where we came from to know how we're getting into. Does anybody in here work with carbon dots or graphene particles for drug delivery or substrate attachment? Highly recommend. We'll get to that next. These were things that were in the past, and now we're talking about what's coming into the future. Q-dots originally came from solar panels. They're just an inorganic semiconductive particle. This here is showing you what molecules will, as a core material, will create different kinds of emissions with different colors of emissions. So they're tuned by wavelength based on the core. None of this is the problem. Like, this is just total inorganic chemistry, understanding how to best harvest the sunlight in a solar panel, or in like a tablet, because your finger is conductive. The biggest problem with nanocrystals is the insolubility of the core, how hard it is to functionalize the surface of it so that we can even attach it to antibodies, multiple antibodies attached to the Q-dot, often cross-linking two antigens together. PEG is the problem. PEG is always the problem. If you think about it, PEG is in our makeup. PEG is in like everything. We're just gonna have like rivers of PEG at some point, because it helps something become miscible to an aqueous environment, just like our makeup is in our bizarrely polarized environment. But the problem with PEG is that, yeah, it makes it soluble. Yeah, it gives it carboxylates to cross-link my antibodies to, but it makes the particle much larger than it needs to be. The Q dot cores are actually super small. They're only about 5 micron. When it starts to get functionalized, it becomes like 3 times larger. And that's where our problem lies. We need to figure out how we can make a material that insulates the core, just like the casing on a plug. What do you call that? An electrical plug. You know what I'm talking about. We need to protect it from the environment so that none of those ions get leached out of the conductive circuit. And that's what this is trying to do, is isolate it, insulate it, protect it, and have it be able to be cross-linked. So independent of that, these have a ton of problems. They still have some place in spectral cytometry, and they have a definite place in microscopy for a lot of their ability to not necessarily be photobleached. I have photobleached them. I took them to a course called AQLM and told all of these like photophysicist kind of people that you couldn't photobleach them. And they're like, let me try. And they literally, I think, like burned a hole through the slide. But that would not be under like the normal conditions. So graphene particles, though, there's a bunch of companies working with graphene, and it's not just to make them into fluorophores. Theoretically, we can use them to integrate capture molecules in order to load them into a sample of patient blood, for example, and have them capture things, use the integrated capture molecules onto the lattice of the graphene particle. And by capturing the analytes, they can become fluorescent. So they're fluorogenic. They can have fluorogenic assays all within this lattice because we can construct this with precision because we know where we're building it like little building blocks. So a really great paper. There's a lot of good papers. But if you look at carbon dots or graphene particles, it's actually the same with the DNA as a polymerizing agent. That's also like a lattice structure for polymerization. You can see people just trying to draw because they can control the structure. People will just like draw shapes and image them through EM and be like, look what I made. Right now, they're often used for bioimaging and drug delivery as well, because I can have them as a sheet, I can have them as a circle, I can have them as a tube. I can load them with different, maybe hydrophobic chemotherapeutics and deliver them directly to a tumor because it's functionalized. Or I can actually use their fluorescent characteristics themselves, load them into a tumor and photoablate that tumor. There's just like innumerable, as a substrate, as a lattice, not just as a dye, it has massive potential for the future. You can sometimes, they're also called, I think, CPNs. I don't know if those are carbon actually. So the brilliant dyes were definitely a big advancement. You will notice that I put the patent number because nobody needs to get in trouble here. All of this is publicly available, and this is the optical unit that comprises the brilliant violet dye. And that's kind of what I'm talking about, is each of these needs to be independently able to express itself without being interfered with by its neighbors. So however many of them I have, the brighter the molecule is. We used to call it a lightning rod. And that's because normally two molecules that are close to one another, they still require the ability to conduct energy between each other. And that means that you have lights on a Christmas tree string. You need to get between, you need to be able to conduct energy between each of those light bulbs, rather than each of them having to be excited by the laser individually. Does that make sense? It's all just about efficiency of conducting energy. So they were a really, really big deal, obviously, but it took us about 3 years to realize that they non-specifically bound, because I had somebody up in Oregon who was like, I don't know what to tell you. This population is shifting. And I'm like, I believe you. I've just never seen it before. So I went through and did a kind of comprehensive 1 by 1, 7 by 1, like just kind of tested out the extent to which the non-specific binding was occurring. If you think about a brilliant violet dye, think of like a jungle gym ladder. That a brilliant violet dye is shaped like a horseshoe. It's not exactly linear. You can think of it like one of those jungle gym ladders with balloons attached closely to the rungs those balloons are the peg. But if I took two of those ladders, I can make them want to basically bind to each other. Does that make sense? And the balloons get pushed off to the side. I'm a visual person. That's how I like to think about it. So the peg is not helping that situation, which is why we need to use the Brilliant Buffer. If they're willing to give it away for free, that's usually a good indication you need to use it. And all it is doing is putting a wedge in between the fluorophores' ability to bind itself. And it's only the core polymer. It doesn't really have a lot to do with what tandem you're using or anything like that. It just is brighter or dimmer depending on that. But that is definitely our biggest problem with the Brilliant polymers. But it was the first place to start to get us off of Q dots. So I think we did a pretty good job. But you can't lyophilize them and you can't cocktail them. And the future of flow cytometry is going to be in our ability to make all of these panels for you. So I had to figure that out the hard way by trying to lyophilize it. And my signal was so bright that I had baseline restoration where your negative sample is brighter, like your unstained sample is actually brighter than your stained sample, because the stained sample, the instrument thinks that your background is out of control and it doesn't understand why it didn't calculate it correctly to begin with. Those were the days. There's also going to be massive limitations on taking these polymers themselves, the core donor, into the near infrared, because as the difference between the BUV and the BV is going to be the thinnest polymer. The BV is going to be a little bit thicker. BV 510 is going to be even thicker. Brilliant blue is going to be even thicker. You can't actually get into the brilliant near infrared because the molecule would become so thick and wide that you'd basically have a square. You wouldn't be able to get it into a long polymer. So why some of these new things are really important. This got us to a certain point. I do want you to take notice of the star bright polymer dots. I don't think that they've completed their R&D on these molecules yet, because this is concerning right here. That's not necessarily good. Those are channels that these are fluorophores that heavily assault other channels. So having them be that bright is not necessarily advantageous. They're also not showing the spill back into the donor channel. But I do think that they're going to fix a lot of our problems when it comes to polymers. For example, you're thinking of it like this long molecule. Imagine that you just started folding it back on itself, like a ruler, like those foldable rulers. It started folding it back on itself so that its likelihood to non-specifically bind with itself is taken away because it is associating with itself. You're, what's the right way of putting that? You're like masking or neutralizing a lot of the non-specific binding characteristics by making it into a dot instead of a linear molecule. It also would let you make it possibly have slightly better transfer efficiency. These often have less spillback and tighter spectra than a lot of the other polymer dyes. This is who invented it down at the corner here at WashU. Highly recommend the papers. But what it would allow us to do is not have to use buffers because it doesn't have the same propensity to non-specifically bind. And the likelihood is that we'll be able to lyophilize it. It's in the hands of Biorad, so it depends what Biorad wants to do with it. But it's definitely starting to expand our selection of polymers off the UV and violet. I've also started using the NovaFluors quite a bit because I was with Thermo for the last year. I think they're some of the coolest because they are just, I know that we keep using that term tunable, but I think you have to go back to how difficult it was for us to control fluorescence before. So this is, it's also, depending on who is making it and selling it, it could be either single-stranded or double-stranded DNA, where the nucleoside is switched out for a fluorophore. So we can build these dyes with a DNA synthesizer. And we know exactly the distance between any two dyes. We know exactly the sequence that's in between. So we can hybridize elements of them together. You can put them into solution and they'll self-assemble based on those hybridizing units, which is what they do for the NovaFluors. They call it a phyton. And they just make it into this cute little tertiary structure that has all these dyes embedded. But that means that we know the distance between two fluorophores. So when we make a tandem, we can tune that spectra, we can tune that brightness based on how big it is or based on the placement of those fluors. That's why we'll get closer to 100% efficiency of transferring energy, because we'll be able to control the angstrom distance between two dyes in order to achieve more efficient frets. Don't be scared of these. There's a few, if you want to talk to me afterwards, just like any other fluorophore, all of them are not perfect. All of them are not necessary. Some of them are really, really great. My favorite is the NovaFluor Red 700 because Alexa 700 sucks. It does require a blocker as well for the cells because monocytes see DNA and they're like, hey, you look like something I should eat. They're doing their job. It's not exactly nonspecific, but it's an unwanted nonspecific binding. That's my dogs because I miss them when I travel. So I do want to say though that at FluoroFinder we are making this intelligent, like actually intelligent panel development tool by giving weights to different choices that we make and using our same process. We're going to be going into beta testing for it in the next month. So if anybody is interested in testing it out, it's part of our grant submission that we need to have like a decent number of people kind of compare what they would have chosen. And there's a lot of right answers in panel building, right? If I use an A5 with like 1,000,002 detectors and I'm only doing 8 colors, I can put them anywhere, right? So, you know, we do kind of have to bring it into realistic, you know, constraints. But if you're interested in that, I'm going to give you my card after. I'm going to be hanging out with the Slingshot people at their booth if you want to come and talk to me after. Thank you. Does anybody have any questions first?
Yeah, well, we have, so if you want to ask questions, we have 2 mics on either side of the auditorium here, and we do have some online questions that are put out. Or do we? This we should ask. Oh, no.
Oh, there you go.
It's early in the morning. Well, hold on. Let's see. Does anyone have any questions for Kelly? Why?
Yeah. No, I can hear you.
Well, just repeat that.
Okay.
Question.
No, you know, you're completely right. That's exactly how I see the situation. So the question was based on disease type, and the kind of the autofluorescence and the ROS and the metabolic issues that are going to come with that particular, you know, the sensitivities for that type. How do you choose fluorophores around it? And the way that I like to think about it is my deep red channels, like even my APC size 7, believe it or not, they are going to be some of my cleanest channels for a variable expression of something that I need to not have perturbed by anything that's endogenous or different. If I look at the difference between a cancer cell and a normal cell, the likelihood that they'll have differences in endogenous autofluorescence in that deep red channel is minimal because nature doesn't usually create molecules that are that large. There is this possibility that they'll be under high oxidative stress. So you might want to consider that in your sample prep. Maybe having something like other proteins around to help scavenge and, you know, kind of be around to protect the fluorophore by giving the ROS something else to go after. You can also have all sorts of things in solution that are going to eventually be washed away before. But I think the likelihood of you seeing that in the few hours or minutes or whatever that it's going to take to actually stain your sample is unlikely. And as long as your control is done under the same condition, then even if it does break down just a little bit, you'll have the control as an appropriate reference.
We have one online question for you, and then I have it. So can you contrast again the red versus blue spectrum floors from your slide in the beginning? Red versus blue spectrum floors in the beginning slide.
The beginning slide. This one maybe? I don't know. I don't know what the question's asking me. Okay, yeah. So that was actually why Alexa floors were made. was that the deeper red that we wanted to take a molecule like Texas Red or Sci5 or Alexa 700, all those kinds of structures, Sci5.5, that one is, we needed to sulfonate them. And that sulfonation would help keep them soluble as the structure grew and became more insoluble. None of, like looking at these here, Sci7, Sci5, Sci3, you can see how nonpolar they are. And we are not a nonpolar environment for these dyes. And it'll end up just quenching them because they're not able to conduct the energy efficiently. So that was kind of the biggest deal, was just to be able to get them more soluble. I think that's, was there more to that question then? Okay.
I did have one question. So this is a question, this is more of a kind of a, you said something interesting on the beginning when speaking about homofred and in particular the DNA dyes. So this is something that's I've never totally understood, even though it's been in my business for years upon years, which is hooks. Hooks for the side population for stem cells. You would see this far red emission, and I've never really fully understood it. So is that?
Yep, that's what that is. Wow. Because the DNA is holding the nucleic acid stain rigidly and within a certain distance because it has a site-specific selectivity. And so because of that, they're just hanging out and they're like, it's hard. They can't not. The only reason that you're not seeing the redshift on everything is because you only have a fraction of them that are close enough in proximity to be doing it. If you saturated the DNA, you would see that shift and then you'd see it go black.
Hence why when the stem cells themselves pump out the dye,
exactly,
you actually start losing that. And, wow, that's really cleared that up. I appreciate that. All right. Well, thank you so much, Kelly.
Yeah.
Please, everyone. All right. So.
So our next speaker is Nico Loof. He has over 10 years in flow cytometry experience. He's currently a senior applications scientist, multi-omic specialist at BD Biosciences. Between 2010 and 2012, Nico served as the flow cytometry SRL manager at Baylor Institute of Immunology. In 2012, he joined the UT Southwestern in Dallas as the director of the Moody Foundation for Flow Cytometry facility, serving over 120 laboratories. In 2021, Nico moved to industry and joined BD FlowJo. He's a recognized member of ISAC since 2010, a former member of the ABRF Flow Cytometry Research Group, and has been a part of the FlowTex organizing committee since 2011. Please welcome Nico.
Thank you, everyone. Great talk, Kelly. I really appreciate it. I hope I do as best as you did. So we're going to talk about the panel design tools that BD offer. So before we go to the tools, let's go to the fundamental of the panel design. To be able to design your panel, you need to know your fluorochrome. So you need to know the brightness, the resolution of your fluorochrome. You need to know the spillover and the spread of your fluorochrome. And also you need to know your biology. And that's where you as scientists, when you come to the flow core, you come into play. You have to tell us what's the antigen density, what's the correct expression in which cells, even if we all have immunology background, we don't know everything and we count on you on that part. And after, we need to know the instrument. So of course, the instrument configuration and the sensitivity. So BD offer tools for all these different fundamentals. So in FlowJo, you can calculate the spread matrix to know the spillover spread. You can use the spectrum viewer to know where's the excitation and emission point for each fluorochrome. Then you have the BD interactive cell map tool, who's really telling you on which cell type, what marker are expressed, and really can go in detail. You have also the BD clone comparison tool to know which clone to pick and what's the difference between each clones. And after, for the instrument, you have, of course, the same thing, the spectrum viewer, and make sure that you go to your code and know the instrument better than anybody else. And since fall, BD released the BD Research Cloud, and BD Research Cloud cover everything that I just mentioned. So before we jump into BD Research Cloud, I wanted just to mention two tools that I think are underused and are really important when you design your panel. And first is to calculate your spread matrix. So in Flojo, into the composition platform, you can calculate the spread matrix easily. You have two types of spread matrix, either the spillover spread matrix, which basically we normalize the fluorochrome brightness and know the spillover between each detector. And you have the total spread matrix. And the total spread matrix we don't normalize. So if you have a bright fluorochrome, it's going to take in consideration the spread from that bright fluorochrome compared to a dim fluorochrome. So calculate your matrix is going to give you which fluorochrome bleeding into what, and you can preview those data, and of course you can export those data. Best practice is to run, I'm sure your core probably have done it, all those CD4 marker in every different fluorochrome. You have the SSM of all those for that instrument, and you can pick the reagent based on that SSM. When you run your panel, just calculate the TSM, total spread matrix, to exactly know, based on your expression in each marker in your cells, what's bleeding into what. The second tool that I think is underused is the BD Interactive Human cell map tool. And it's an online tool designed to help and plan the first flow cytometry experiment. And can know the immunocell hierarchy, the immunophenotype. You can access to pre-designed flow cytometry panel and data, and a link to any BD technical and scientific. All right, so now let's go to the heart of the subject. Today is the BD research. So what's the BD Research Cloud? It's an online ecosystem that's supporting flow cytometry study. So when I joined BD, the idea is like, I was like, okay, BD has all these different tools, but they don't connect to each other. So let's try to find a way to connect all these tools together. So in BD Research Cloud, you can, of course, upload your instrument. It can be, of course, a BD instrument, it's BD, but you can put any instrument that you want. You just have to manually enter your instrument. You can create your panel using reagent from the BD catalog, of course, but also you can bring any custom reagent into the instrument. So if you're using any competitive fluorochrome, you can bring them into, use them into the BD Research Cloud. And we're going to try to improve that. We don't have everything right now, but we're going to try to improve and really bring all the fluorochrome that are available currently on the market. You can manage your inventory, so what you have in your lab. So you always open the fridge, say what you have. Oh, I have a CD4P, I'm going to use that today. Not the best choice, but you can do that here. And you can, of course, see the spectrum view. The first advantage or tool in the BD Research Cloud is your workflow. So you can create panel, you can create experiment, and it's really play the role as a lab notebook. So you can say, I want Carla to do this project, I want Mike to do that part of the project, and as soon as it's done, it can give me a notification telling me that's me now that I have to do my work on that project. You can create your DIVA experiment away from the instrument, so you don't have to spend time on the instrument and waste time on the instrument. So it's going to allow you to really enter your team name, plate names, all the information, all the metadata that are important for your experiment. And after when you run your experiment, you can store all those data on the cloud and directly have access to them from the cloud to Flojo. So how to create an account in BD Research Cloud? Just go on our website, bdresearchcloud.com. You can use your Flojo portal account if you have one. If you don't, just click on the create account to have access. BD Research Cloud is free. So we have different tiers. So for individual user or for small group, it's free. If you want to go to big institution, of course, you have a cost associated to it. All right, so the structure of the BD Research Cloud. In the BD Research Cloud, you have groups, and group represent people or user working together on the specific project. when you invite, when you have a group, you need to have an organization, and this organization can be in different thing depending on what you want, and each group will be assigned to an organization. Then within the group, you're gonna have the project, and each project is really where you're gonna have your data, your protocol, your panel design, and everything. One user can belong to many groups. You don't have to be assigned to only one group, and a user that's assigned to one group can see all the project from that group. So also be careful if you have confidentiality. If you're part of a group, you're gonna see everything. There's also no limit of number of groups, so create as many groups as you want. So as I mentioned, this organization can be used in different structure. Either you put the institution as an organization, and after the group is a different lab or department, and after you see the project, or if you're an individual lab, can be your organization is your lab, and you can have the group based on the different project within your lab. This organization really allow you to collaborate in different projects. So it can really organize the colleague into group, assign tasks and step and do that, and a lab notebook and custom field to capture metadata. It's also used as a lab notebook. So really allow you to store your data, put notes on everything that's happened during this experiment, track reagent inventory, and organize instrument in the custom workflow. So back to the panel design. So as I mentioned, we can store instrument configuration. So we'll only give you the reagent that are available on the instrument. We're gonna ask you to assign what's the expression of this marker. You can assign the markers, but also directly the type of cell that you want to analyze. So instead of saying I want CD4, CD25 and everything, you can just say I want naive B cells or CD4 T cells, and we're gonna assign directly all the marker commonly used to define those population. Then we're gonna have the list of reagent, and you're gonna be able to pair this reagent. And of course, because we want money, we're gonna also provide an easy way to order those reagents directly. So as I mentioned, you're gonna be able to create experiment. So you can, in the new version that's coming soon, you're gonna even be able to assign voltage for each marker. So it's just gonna be a random voltage, but some people like to define the voltage before, so we're gonna give you that. But you're gonna be able to put your keyword. You can be able to assign the fluorochrome based on the panel. You're not gonna have to enter anything because we know which panel you design. We can import all this information within that experiment. And when you go on the instrument, you just have to connect BDRC to DIVA, and all this information will be automatically assigned. Then we're gonna go on FlowDraw after you run your experiment, and you want to open your FlowDraw experiment here. So you're gonna be able to directly from FlowDraw open the BD Research Cloud, add your FCS file within FlowDraw, analyze your data, and save this workspace or analysis directly back to the BD Research Cloud. All right, so enough talking. Let's. I'm gonna show it to you. Hopefully internet work. I'm gonna have to share the screen. Yeah. See. I'll put my credential. So when you get to right, that's the zoom button. It's gonna move that away. All right, so when you get to the BD Research Cloud, you have your name, you have your organization here. I'm part of many organizations. Just gonna go to mine. And you're gonna be able to create your panel. So already have an instrument assigned, already have everything part in the different group. So I can really I can directly go to create my panel. So go create my panel. I can name it. They can assign my project, and I can start from scratch. Here I can choose my instrument. Okay, so you have the BD default instrument, but you can have any instrument that you define previously, and you're gonna be able to select. You're gonna just select this SR2. You can select which as target you want, so human. And here I can just go for CD4, or I can just say I want T cells. And if I say T cells, it's just gonna give me all the marker, for example, for the CD8 T cells. Load more and see even more T cells. Let's take the T helper, add population, and give me all the marker for those. Can assign the intensity of those marker on the expression of those marker on those cells, and I can complete. Then I just have to pair. So I can say organize them by antigen density, organize them by flow chrome availability, and you can pick. I want CD197 with BV421. I don't know if it's a good choice, Kelly. You tell me that, but I skip that. And I can keep going and assign everything. And at the end, you can, depending on your organization, you can either order it directly or you can just request a quote from your BD rep. So that's how you design the panel. We're not gonna give you too much advice there. That's, of course, in the pipeline in the future. But right now, it's just basically a reagent. Back to phase. You can create. Don't know. I'm doing with time. I think I have 2 more minutes. So you can create experiment. I think that's the beauty of the BD Research Cloud, to be able to create your experiment away from the instrument. So you have free time on the instrument. Go are busy, the instruments are always in use, so you don't want to waste time creating your experiment in front of the machine. With BD Research Cloud, you just can create your experiment there. So I'm just gonna open this experiment that I just created. You can select if it's a plate or not. If it's not a plate, you just click continue. You can add sample, add team. You can select. There we go. All right, so I can select my panel. I can doesn't know which instrument I can apply. Can show all the markers, so directly have all the marker. I can add my specimen, my tube. I can continue, and I can keep going like that all the way to add my keyword, and at the end, creating my dot plots on it, and just export it directly to Diva. And I think my time is stopped there. And anyway, I don't have FlowJo here, so I cannot show you on FlowJo. But I'm happy to show it to you at the booth, or stop by, and I'll show you more in detail. Thank you.
There was one question. Sure. There's an online question that whether you could actually utilize this to show the spectral, the total spread matrix between the floors.
So in FlowJo you can, not in BDRC right now.
Thank you so much. Thank you, Nico. All right, All right, everyone. This brings us to our first break, coffee break. We have a half an hour coffee break. It's obviously it's across the way for the people in house. You just go, you just kind of cross over to the next area where there's vendors, food. There's still kolaches left from this morning, and please go and help yourself. Please talk to the vendors, like you said. I want to say thank you so much to them for supporting us. And for those online, to get your coffee, go into your kitchen and open your fridge and grab your snack and grab your coffee and then be back here in a half an hour.
Good morning. I hope everybody enjoyed their coffee. We're going to restart and I will introduce our next speaker. So our next speaker is Deanna Bonilla. She has a PhD from Texas A&M and did a postdoc at Baylor College of Medicine. So she's been here in the Med Center for a long time. She's an ASCP accredited cytometry specialist and has been working in the field of cytometry for over 20 years and has an extensive variety of applications that she's worked with. She actually worked in my lab at MD Anderson for a few years and then went on to work at the immunotherapy platform there at Anderson and is currently an ISAC SRL emerging leader and highly involved in ISAC activities and task forces. She currently works as the Global Scientific Director for SciTech Biosciences, and she is going to talk to us about how to build a successful panel and 40 color space.
Good morning, everyone, and thank you so much to the committee for the invite. It's always great to be here, seeing friends and talking about flow cytometry. This was a difficult task for me because the request was to put together a talk that was going to go through the fundamentals of panel design in the context of a 40-color panel. So it's a low complexity, high complexity, but good thing it's the rules that we apply for panel design are going to be useful for anything from 5 colors to 40 colors. So definitely doable. And the other thing, it's panel design, no matter what instrument you're using, the rules are the same. So definitely, if you're not using spectral, which is going to be the main topic of the talk, the rules still going to apply if you're using conventional cytometers. And everything starts from this publication, is the OMIC 69, an optimized panel. There is so many of these optimized panels published in the cytometry journal, part A. This is one of them, and it was done on a spectral cytometer. It was, the goal of this panel was to optimize 40 colors together for PBMCs or for blood. And the publication is so useful, and that's why I put it in the first slide. Let me see if we can get rid of that. There you go. And it's useful because it's going to show you more details of what I'm telling you today, telling you how the panel was put together, how the fluorochromes were selected, but also, which is more important, it's how the panel was validated, how we defined that combination of fluors was indeed successful, how we incorporate then controls, acquire samples to make sure data is high quality, which is the ultimate goal. So in general, panel design, once again, no matter the complexity of the panel and no matter what technology you're using, needs to be an informed process. And let's divide it into 5 steps. We start with the biology, similar to what Nick was presenting. You need to first know what's the system you want to use, what cells you want to characterize. Define that if indeed flow cytometry is what's going to give you the best readout. And once that's completed, focus on yourself. What markers will define the lineage of that population? What markers will answer your scientific questions? And what's the level of expression? So we'll go one by one just to give you a general idea. Another important part of that panel design process is understand the instrument. What's the configuration? What lasers are present? What filters are there that's going to define what fluorochromes you will be able to characterize? What fluorochromes are available commercially? What fluorochromes you have in the fridge? What fluorochromes you decide to put together? And then the design part, knowing all these parts, the antigens, the instrument, the fluorochromes, how you put that together in a panel that it's high quality. And finally, and definitely the most important one, the optimization, when you go and test every single reagent, single color controls and the multicolor sample and define if that combination on paper, it's really a good one based on the quality of the data. So if we go step by step, biology first, we need to define what markers will be in the panel, what's the level of expression of those markers, if they are co-expressed or not, and we will go in detail on understanding why that's important. From the biology, what's the scientific question? My population of interest. From that population of interest, what markers will define that population? Which markers will allow me to exclude other populations? What markers will be important to define more functionality, like memory markers, exhaustion markers? And what markers are critical to define what's the question? Let's say you're interested in inhibitory receptors out of an NK populations, and all those tertiary markers that are not highly expressed, markers that you need a control or a way to induce expression in order to make sure the panel is working appropriately. Part of this, knowing the biology, it's defining what markers will be expressed within the same cell at the same time, because that's also part of what we use when selecting the fluorochromes. So let's say you are interested in blood samples, PBMC, so you care about all the CD45 positives. So that's a marker that is going to be in every single cell in your sample. Then you go down multiple subsets. Let's say you're interested mainly in lymphocytes. So you know CD45 will be also expressed or co-expressed with CD3 if you care about the T cells, or CD3 will not be present, one of those exclusion markers for NK cells, but knowing that in NK, in humans, you will be finding co-expression with CD56, depending on the functionality for that NK subset, you will see it dimly expressed or with higher expression. 16 probably present in most of them, CD2 in all of the NKs. So a lot of considerations on what markers will be present in the surface of the cell at the same time, and then select the fluorochromes based on that. So you could do it using this tree of information of positivity and negativity of the different markers, but you can also do it through your gating strategy. So if I care about NKs, then probably my first gate out of all the markers in the panel is going to be excluding the positivity for CD3. Then defining CD16, CD56 for NKs, where you will see multiple subsets with different intensities for those markers. So those two markers are going to be critical. There is a chance for co-expression. So the fluor assignment will be highly dependent on what you have in your gating strategy, especially if you care about this upper quadrant, the right one, where the co-expression will be assessed. And same with every marker in the panel. So here's where it comes. If you have 5 markers only, then you will go to only positivity or potential positivity for one of them. But the same will go for 40 or for 30 out of the, excluding the lineage markers, depending on what you have in the panel. So this part is super critical. What markers you wanna measure? What's their level of expression? How, if they are co-expressed or not? And what's the gating strategy that you plan to use, even if it's just theoretical? Again, because selection of fluors will highly depend on antigen density. Then know the reagents. It's not only detecting CD4, and back to what Nick was saying, it's what's the clone that will allow you to detect CD4 in a more accurate way, or based on your staining protocol, or based on the conjugate you plan to use. Some clones work better with certain conjugates or fluorochromes. So knowing that information is highly critical. There is a lot of websites that provide information on antibody validation, and that includes what clones were used. So important to know that information. Also, the experimental conditions. Do you need to fix your cells? Do you need to do permeabilization to be able to access intracellular targets? All of that is going to also change how clones are performing. So important to go to vendor websites and understand what characteristics of that clone are defining there. What's the dilution recommended? What are the characteristics in general? And also a lot of literature, which is critical in this initial step, and there is a lot of publications citing Cytometry Part 8 journal, publications where the populations are described, more scientific paper, more of a technical one, the OMIPs, like this OMIP 29, that it's only focused on NK cells. And finally, when I was saying that you need to know how much an antigen is expressed, sometimes we don't know, or you don't have information on that. So there is also publications like the CD maps back in 2019, that characterize all the CDs from 1 to 11 to 100, and tells you in different subsets out of hematopoietic cells, what's the expression of CD1, and in dendritic cells, what's the expression of CD100 in T cells. So it's a useful tool to understand if a molecule is highly expressed or not, if you don't have any previous data to know. But if you do have previous data, anyone in your lab or in the literature, this is an example of an NK panel, is internal data from Cytek. So from here, just looking at the plots, I'll be able to tell out of the cells that are CD3 negative, if I want to know what's the expression of 16 and 56 unstimulated cells, untreated cells from a normal donor. Now I see that I have multiple populations, right? Some of the cells are 16 positive, dim for 56. Some of them are double positive, a little bit brighter for 56. And some of them are only 56, low frequency for this population, and they are negative for 16. So we want to make sure that your panel allows you to characterize and separate well the dimly expressors for 56, the intermediate ones, and the highly expressors. So you need a fluor that allows you to separate them well. That's why understanding what's the expression pattern is so critical before you go and assign fluorochromes. Of course, these conclusions here depend on what fluors were originally assigned to this previous data, but important that now I know for CD2, almost all the events that were CD3 negative and 56 positives are going to be positive for CD2. So CD2 is going to be in those cells, highly expressed, and it's going to create issues we will see later based on the fluor that I select for CD4 in terms of spread added to others. And that's so critical for panel design. And if we go further on this particular data set for NK cells and NKT cells, which are going to be the CD3 positives, now I see how different the expression levels can be depending on the marker. For CD56, almost everything was positive. Same thing with CD2. Right? But now I get markers like CD62L. This depends also on how the cells were treated, on the conditions of the experiment. But most of the events are negative. So knowing that is critical because one, I know I need a fluor that allows me to separate well those negatives, but also knowing that those negatives are dimly expressed will not be such a problem in terms of spread for others. And the same with other markers, right? Markers that are dimly expressed, markers that are more of a secondary level of expression. We will see the categories later. But so critical to look at previous data and understand for every single marker, how things look and how I need to define the fluor selection. So in general, for antigens, we can categorize them in three groups. Primary antigens, and if you look at data, let's say a histogram, you will see negative events and positive events. So that's a on and or off expression. Tends to be the case if we talk about T cells for CD4, CD3, CD8. You have it or you don't, but you can clearly separate them. Secondary expression tends to be the case for memory markers, where you see more continuous of expression. So you see the negatives, but you see this population in between and then the higher expressors. But most of what it's in between is that transition between memory status or population. So you will see it on CD45 array, CCR7, CD62 ligand, HLA-DR. It's, again, not on and off. And what you see also is that the amount of molecules that are present in the cell are going to be lower than compared to a primary antigen. And finally, the tertiary ones, the ones that are not highly expressed. You will see less events, less frequency in that population. For T cells, tends to be the case for most of the checkpoints, including PD-1, for CD25, TCR gamma delta. So if you see at a histogram, look at a histogram, but you will see most of the events are negative, and you see most of a shoulder, easier to see on a dot plot or pseudocolor plot. It's just that the population that is positive is really low. And on top of that, the amount of molecules of CD25 that are present in those cells is also lower. So it's more difficult to detect them. So, and this is critical because we will pair in panel design the level of antigen expression with the fluorochrome brightness. Why? Because for CD4, you see the separation. This is a bright fluor. Anyway, so it depends on the fluor when you look at the data. The easiest way to compare across markers would be using the same fluor, let's say PE. The point is that if you want to distinguish what's positive for CD4, you don't need a bright fluor because you can easily see that separation. So we can use dim fluorochromes for these primary markers and for the tertiary markers, since it's so difficult to see it because it's low expression. you want to reserve your brighted fluors in that case. And anything in the secondary can be assigned. We will see the steps. So now moving to the 40 color panel. These are the 40 markers. We selected the clones based on previous literature, based on performance, based on the staining protocol that we wanted to use. We also, every single marker, we knew it had a purpose within the panel. Either define a population or go and add additional information in terms of functionality. This is the tree for those 40 markers. As you see, lymphocytes, monocytes, out of the lymphocytes, T cells, non-T cells, a huge characterization, not only again in lineage, but also functionality for those populations. But this again will apply to a 5-color panel or a 40-color, as just knowing what markers will be there. If you care about cytotoxic T cells, let me see. CD8 will be then on those cells. If you care about memory status, you might or not have the presence of CCR7, CD45 array. And then you could have negativity or positivity for so many other markers. So how you assign fluors, again, it's so critical. And as well for other populations, like in this case, the PDCs and the MDCs, and how we make sure we define them correctly, how we make sure we exclude populations that are not part of that subset, and how we go additionally on characterizing how those cells are working in terms of functionality. Step number 2. We know the markers well. Now we need to know the instrument. What's the optical configuration? Is your instrument performing well? And finally, you QC the instrument before you run the samples to make sure you're getting high quality data. I'm going to focus, because of the 40-color panel, on a Cytec instrument. This is an Aurora unit. It has 5 lasers. Those 5 lasers are specially separated. They reach the flow cell at the same time, but the cell is going to be interrogated at different times. All that information will be collected. After the cells are interrogated by the lasers, we collect the light, scatter light. So it has a Fourier scatter, 2 scatters out of the blue and out of the violet, and a lot of fluorescence detectors. The detectors are organized in modules. So after, for example, the UV laser interrogates the cells, we are going to collect all the emission out of that interaction, and we will see how that emission is organized. Every module of detectors will have detectors organized in sequential order. They are collecting different wavelengths, and this is the bandwidth. Anyway, the key point for spectral is we collect the full emission profile. So what you will get at the end from this full spectrum profiling is one plot. The plot is called the spectral plot. It tells you the intensity of the signal, how many photons reach the detector and were converted into electrons, and then that signal plotted in this graph. And on the x-axis, you will see every single detector in the instrument. For a 5-laser unit, you will have 64 detectors organized in modules. So again, after UV excitation, this is all the light, all the light that was collected by those detectors. We see how some of those detectors get a lot of intensity. The highest for this particular fluor, it's violet 14. That's where we get the best emission, or the highest emission, and the best excitation by the violet. But we also see that all the lasers were able to excite this molecule. And that's why we get higher intensity in the y-axis. And kind of one of the differences between a spectral and conventional, right? That instead of having one dedicated detector per fluorochrome, you have 64, again, versus conventional, where you mainly capture the peak of emission. So why it's critical to have full spectrum profiling when you talk about a 40-color panel? It's because since we capture all the emission across all those detectors, what we have for each fluor is a signature. The peak for 2 fluors might be the same, or could be the same, but the whole ups and downs of these signatures can be different. So that allows us to distinguish them as unique. And that's kind of the first thing you need to have for a big panel: unique fluors that will not be giving you any confounding factors. For the 40-color panel, you will see, if you read the publication, combination of fluors that traditionally by conventional, you cannot use them together, 421, super bright 436. If you look at the signature, it looks very, very similar. They have a very similar excitation emission pattern. But since we have this short bandpass filters for each detector, we know that 421 emits the highest at violet 1 detector versus super bright 436 at violet 2. So now we can use them together in the same panel with careful assignment of markers. And the same applies to other combinations, APC, Alexa 647. You see how similar they look, but the tiny differences allows us to use them together. 515 and FITSI, PE and yellow-green 584. So a lot of combinations that allow and this 40-color panel was published 3 years ago. So many things, and you will see a slide on that, have changed, and a lot of development on reagents as well. But at least 3 years ago, this was kind of the best selection with the fluorochromes that were available. Step number 3, the fluorochromes. The second most important player, how you select them, right? 4 things that are critical: that they are unique, how bright they are, and how you will assign based on antigen density. The spread. So we will dedicate a few slides to understand what a spread is and how you try to minimize it. And finally, how to do the assignment based on all those characteristics. These are the fluors used for the 40-color panel. And you can see this type of graphs if you go to Cytek website for the spectral viewer. So as you see, the first thing is at least that they have different peaks, emission peaks. But for some of them, like PAC Orange and Brilliant Violet 570, you see the peaks are exactly at the same detector. For PE and Yellow-Green 584, exactly the same peak. The critical part is that it's not only the peak what matters, it's the full signature. And their signatures are different enough, even though they're peaking in the same detector, to be able to use them once again together. So there is, you could do it by eye, right? Just creating this plot, but it's really difficult when you go to 40. So we have a metric that is called the similarity index that tells you if 2 fluors can be used in combination or not. And the similarity index is a number that goes from 0 to 1. 0, no similarity, 1, identical. And if you go to that website, we'll give you all the metrics for similarity between each of those 40 colors versus all the others. And you will see most of the numbers are around 0, so low similarity, but some of them, like for example, APCH7 and BUV395, you see highest emission out of the UV for one of them, the other one out of the red. So not a lot of overlap. In other words, they are not highly similar. And that's definitely indicated by the number. You see there is a 0 in similarity, but we also see some blue colors. For example, the one we were talking about, super bright 436, 421. If we look at the emission profiles, they are very on top of each other. We saw it also from the signature. Their similarity is 0.97. So it's getting to a 1, indicates that they're highly similar, but based on our experience, they can still be used together. 0.98 would be like the cutoff. This matrix will also give you at the bottom, the complexity index. And it's a number that with all the combination of fluors that you decided to use, gives you an indication of whether it's a good panel. It's great to compare. If you have 2 combinations, the one that with the lower complexity will be the easier one to use based on how those fluors look. So this is the final decision after looking at brightness and what fluors could be used together. And again, 3 years ago, these were the fluors that were used. We like to use this grid because it's very easy for me to assess a panel if I see all the markers and if I see them organized by the highest excitation, the laser that excites the better, and by the peak of emission. We will see that later. But how do I know which fluor I need to put or pair with CD4, for example? Well, that's going to be based, and this is part of related to a previous presentation, and there's characteristics from the fluor. One of them is brightness. And the way we measure brightness, it's by calculating the stain index. This is the formula. It's mainly taking into account the intensity of the positive population, the intensity of the negative, and also how spread the negatives are, put in a single formula and then get a single number, that stain index. The higher the stain index, the better the separation between negatives and positives. How is the positive population or the intensity of that population affected? Well, how bright the fluor is, but also other things, right? What's the expression of your antigen? The characteristics of the antibody, if it's an IgG versus an IgM, the performance of that antibody, once again, the clone, And definitely depends a lot on sensitivity of the instrument. So if you use highly sensitive detectors, the gains that were used or the voltages that were used for acquisition, how well your optical are performing, if your lasers are well aligned, if in this case, if your APDs are well aligned. So all of that is gonna play a role in how well separated things are in terms of instrument as well, because the negatives are gonna be influenced by the noise, electronic noise, optical noise. So a lot of that's playing a role in how these things separate, not only the fluorochrome brightness, that will depend more on the molecule, how well it absorbs and emits light. But if we keep constant, the same instrument, the same antigen, so we do the comparison, let's say across CD4, we do it with the same clone, we do it with same titer and with same instrument, same gains. So what we ended up comparing is actually just the fluor brightness. So I'm going to show you for the same target, CD4, how it looks if I use PAC orange or different fluors and one that is brighter. So the separation being the same marker, so it's independently of antigen levels of expression. It's less because PAC orange is not that bright. But if I use BYG 710, which is a brighter dye from Cytec, you will see that is much better separation. So you don't need to do this character. Oh, wait, sorry. Let me show you on the bottom, because that's related to the stain index. So if this is, again, low stain index, because there is low separation, and higher stain index because of this situation. We give you this ranking. So you will get it in the fluorochrome guidelines. That tells you, based on the stain index, what are the fluors that are dimmer, what are the fluors that are brighter, and what's in between. Once again, because that brightness will define what antigen I'm going to use to pair with that particular fluorochrome. So we reserve brighter dyes for dimly expressed antigens and dimmer dyes for tertiary markers or markers that are highly expressed. But it's not only about how bright the fluor is. It's when you put multiple fluors in combination, they are going to influence each other. Not only fluorochrome interaction, but how much light that fluorochrome, how much spillover a particular fluorochrome is adding into others. Spillover is something that you could correct mathematically through compensation or unmixing, but still the light that a fluorochrome is adding into a secondary detector, that it's important for another fluor, like being the primary for another fluor, then you will see that then is going to be an effect. In the classical case is PE-Sci5, APC. These are the signatures. PE-Sci5 in this configuration will give you the highest out of the blue module. So blue 8 is where we get the highest intensity. APC is a red dye, so we will see the highest out of the red module, R1. But what we're also seeing is that these two fluors, like PE-Sci5, it's adding light in the red detectors that are not the primary for PE-Sci5, but are detectors critical for others, like the APC. So in the APC detector R1, we get a lot of light from PE-Sci5. And that translates into spread that cannot be fixed, that it's part of the nature of the fluorochrome, and it's going to be reflected in your data. So here it's difficult to compare because the intensity might be different. So if we normalize having both of them the highest at 100, we see that in the critical area for APC, there is a lot of emission from PE-Sci5. And this is how a plot will look like. If I plot APC versus PE-Sci5, single stains, you will see that there is this population that it has a high spread, right? And that spread is not only visually impacting, but also important because now the area where you want to look for co-expression, and that's why it's so critical to know about co-expression, will be reduced compared if you don't have much spread. So APC is not spreading much into PE-Sci5. We were seeing it here. There is not much emission on APC in the critical detector for PE-Sci5, but PE-Sci5 is spreading into APC. We will see it here. We will see it in the data. We see how does this trumpet shape or umbrella shape looks like. And why it's important? Because now my ability to see co-expression is going to be reduced. And it depends on the markers. So it's a highly expressed markers, maybe it might not create any issues. But if it's a dimly expressed marker, we will get into that region where you will not be able to distinguish what's single positive and what's the co-expression with this dimly expressed population for the APC. So that's why it's critical. What fluors I'm going to put in combination for markers that are co-expressed. But again, you could run the samples and identify the spread. It's an issue. It's very predictable by using the signature. You could run the single color controls and understand if you're getting into this situation. But there is also metrics that allow you to conclude from a previous set of data, this using CD4, knowing that CD4, it's kind of intermediate expression or in terms of intensity. So if you have a molecule that is higher expressed, as we saw before, the higher the intensity, the more spread you will get. So if you're dealing with this one, it's not that critical that if you're dealing with a population like this. So many factors to take into account, but we provide you with the metrics. This is a stain index reduction. Nick was talking about GSM by SSM, and there is multiple ways to measure and quantify spread, different formulas, but at the end, all of them give you the same information. If that number that was calculated is low, white in the chart, no impact. So fluor is not impacting others. But if it's red, it's impacting. And we want to get away from those impacting zones if you have co-expression. So just an example. And the way you read this tables is you want to see how much of fluor in the rows it's affecting a fluor in the columns. So let's see C-fluor blue 515, how much is affecting C-fluor violet 420. White color, no impact. And when I look at the plots, that's indeed what I'm seeing, right? This is a single color stain, so you would not expect to see anything in the upper quadrants. The spread of this population is very similar to the negative. It's really tight, so it's not going to impact it. And we see it. We see it from the number and from the data. But if I go to blue 515, it can affect others' eyes, including the blue 532. So if I now look at these two, very different, right? And it's the same 515, it's how much it can spread into other fluors. Here the spread is higher. And once again, why that's so critical? Because this spread could affect your ability to see dimly expressors out of whatever you put on blue 532, and not much issue if what you use is violet 420. So all that information about the antigens, about the fluors, it's now we need to put it together in the design. Fluor assignment, same rules. Usually we start with tertiary antigens because those are the ones that are less frequently commercially available, because you want to reserve the brighter fluors for them. The secondary antigens go next. For secondary and primary, careful about co-expression, because anything in them can impact the tertiary and your ability to resolve the tertiary ones. If there is no co-expression, you can use any bright dye. But if there is co-expression, you want to make sure that that bright dye doesn't add spread, as we saw with the table. Primary antigens, they are usually often available in many conjugates, so they are easier to assign. And now you can go for a dim one that doesn't spread into secondary and tertiary ones. So just to go through the process, these are the fluors we selected for the 40. Sometimes there are decisions before that tertiary, secondary, and primary. Like we knew light blue was a great option for a big panel, so we started with having a viability dye. You know how critical it is to be able to exclude dead cells from the analysis. Also, depending on reagent availability, for the yellow-green, only CD4 specificity was available. Similar, very few for PE Alexa Fluor 610. The PE Fire 810 and APC Fire were being developed at that time by BioLegend, so no many conjugates available. So all those decisions were first in assigning those fluors. Then we came with tertiary antigens. Markers like NKG2C, NKG2A, low express in the system we were going to use. So we went for bright fluors, PE and APC. Secondary antigens, and giving you only two examples because the same applies to all the tertiary and all the secondary ones. 57, we needed to pick up a fluorochrome that was not as bright because it was going to be co-expressed in many of the subsets. And then primary antigens assigned to fluors that are dim. These bars in the chart are indicating the fluor brightness. So PerCP is not as bright. CD45 was a good candidate there. Same thing for CD16. We were expecting a lot of co-expression with other markers, so we put it in a fluor that is far from what's critical and also a fluor that is not as bright. So in terms of spread, it's not contributing much. After doing all of that for every single marker, this is the final selection. And there is a step that we do, and it's called the theoretical QC of the panel. First, because we want to make sure that detectors that are markers that are in neighboring channels, like CCR7, CD123, because these two fluors are highly overlapping. We don't want them to be in or assigned to markers that are co-expressed. So we have one in dendritic cells, one in more toward lymphocytes. And not only neighboring channels like this two, but across lasers, because you can have cross-laser excitation and emission. What we can then do is 16 for NKs or myeloid populations, IgD in B cells, and then try to assign things across subsets so you don't get that spread issue I was telling you about. But again, that was 3 years ago. Now we have an option that it's the Cytek Cloud. And there is a way to build your panel in a more informative way within the cloud. You can create a free account just by logging into Cloud Cytek Bio. Whole thing, it's all those tools that I show you are available, like how similar dyes are, the spectrum viewer, how complex the panel is, what are the brightness of the fluors from dim to bright, what's the spread, the stain index reduction, what's the antigen density, the panel metrics, and what reagents are available. So all of that is in the cloud if you want to give it a try. You have the markers, the fluors. It's going to give you this overlay of the signatures, going to give you the grid. Within the grid, you will also get how similar those fluors you selected are. It's going to give you a chart with the stain index reduction that I show you, a spread way, or a way to assess spread. So that information will be there as well. And it's easy to select and just assign things. The final is that very similar to what Nick was presenting, is that you will get a chance to, well, get all this information in Excel or just to be able to order it. But you will also get a chance to design your experiment from there. So you don't need to type all these markers again when you go to the cytometer. You can create your experiment and start from it. And an important thing from that panel builder is that fluor assignment for the 40 color panel was in CD19, in 2019. The complexity at that time was 53, was high. There were very few fluor options available to build a 40 color panel. But right now we are in a better situation, right? The super bright dyes, the spark blue dyes, the sea fluor dyes, and a lot of other combinations that allows us to go lower on complexity and lower on the issues that between fluors that we could have. I don't have much time, Joel. Awesome. Okay. So perfect, because this is my favorite part, is how you go from that theoretical design to have a panel that really works. And it's all the testing that needs to come after just creating that grid with the perfect combination of fluors and deciding whether you need to change something. First step in optimization is titration. You need to know what's the optimal titer for your marker, for your conjugate, in order to then have a panel that with 40 markers, it's cohesive. Needs to be done, not only for the antibodies, for functional dyes or viability dyes. You ideally test from 8 to 12 dilutions or antibody concentrations, and say nanograms per test. And what you will see here clearly is that if you will reach a saturation phase, so you want to be in this space where you are in antibody saturation, but you don't want to go too high that you are just increasing the background, the non-specific binding. You don't want to go too low that you're not at saturation, because then the staining conditions will impact a lot what you get in terms of positivity or intensity of the signal. So you want to select the titer that way. It will help you define what's the concentration of antibody that allows you to resolve better positives from negatives. Key point on titration, but important to know that it will highly depend on how you stain the cells. So you need to know what tissue, what are the conditions for how the cells were prepped, were stained, and what lot and reagents are being used. And it's so important because if the incorrect antibody titer, you can have, if you use too much, you can have signal that is out of scale. If you use too much, you can just increase the background. if you don't use much, then not getting the proper separation and then having a risk for bad quality in your data. This is an example of the Omics 69. If you go to the publication, you will see all the 40 markers being, or conjugates being titrated, and then how you pick the optimal one based on what you see and also based on the numbers. You can calculate stain index and define based on that. And an important consideration is you want to do all the testing in the same final volume, ideally the volume that you would use for the multicolor sample. So if you have 40 markers, 250 microliters, probably 300 microliters, try to have that as a constant in your titration evaluation. Controls. For both conventional and spectral, you need single color controls. We call them reference controls. And it's, you want to bring one control per fluorochrome in the panel, so 40 controls in this case, but also you want to make sure the carrier that you're using to prepare that particular control, cells or beads, and critical the type of beads, will give you good estimation of the emission pattern for that particular fluor. If you see things like this, where populations are curving in the single stain, that's an indication that your control is not the best, that could be improved. For example, this tends to be the case if you're using beads with certain fluors. If I overlap how cells and beads look in that full emission profile, normalized to be able to compare, I see some areas in the peak detector, they are exactly the same. So it's definitely defining well what BUV 661 looks like, but it's not defining well how, and you will see some differences here between cells and beads here as well. It's not defining well for other fluors. So that's why we see this error in unmixing when you go to the BUV 395, exactly right here, and when you go to APC, exactly here. So that can be fixed by using cells, because cells are going to represent better what you have in the multicolor sample. But it's definitely doable to use beads. Out of that 40 color panel, 30 of those markers use beads. We use beads for, so definitely doable. Just make sure you compare. What's the ideal situation? That if you look at all the permutations N by 1 for BUV 395, that there is alignment between the negatives and the positives, because the correction and mixing or compensation was done properly, that you don't get those populations curving. That's the ideal situation. But sometimes you might see things like this. Again, curving down or up. I think I highlighted it. Yeah, up tends to be in the areas where there is similar emission between one fluor and another, but that needs to be improved in order for the panel to work well. Why? Because you will have, if in this case, you will have a lot of negative events, that's going to look back in the data. And in this case, you will have false positives, because anything that is only spark 550, blue 550, will look as positive for FITSI. So important to correct for that before you start the testing. Important that you look at your fully stained sample, permutations, how cells versus beads look for that calculation of the spillover. And then finally define what's best. Also important to assess the panel beyond that theoretical evaluation is, if you stain only for CD, let's say CD4, only CD4, this is the separation. But let's say you go to the multicolor scenario where 39 more antibodies were added, and now your CD4 looks like this. So there is something within that multicolor scenario that it's affecting your ability to resolve CD4. Things that could happen, spread, right? That fluor is receiving a lot of spread, and that wasn't the positive, but it's translated into the negatives, right? That's increasing the background for the negatives. It could also be steric hindrance, and it's when I have only one antibody binding to the cells, that's an easy scenario compared to when I have 40. And it's also going to depend on how highly expressed your marker is. If it's not that highly expressed, then more difficulties for the antibody to bind to the target. So that could happen. And also unmixing inaccuracy. So all those errors could affect your ability to see the population. Single stain controls are extremely useful. It tells me if my antibody is performing well, if the titer is good, if the staining conditions were not affecting my ability to see the expression, I'll capture that expression using an antibody. Important just to make sure that mixing is working well, compensation is working well, and the quality of the panel. And I'm going to skip this one because I want to, it's very similar to this. Let's say you have a single stain and a multicolor for 127. You expect the gray one, it's the single stain. You get good separation between negatives and positives, limited by the fact that 127 is not highly expressed, so you will not see that clear separation even using a bright fluor. But when you go to the multicolor with all those other antibodies, what you get is the separation is affected. The positives get dimmer. So how you can control for that? You can change the fluor, or you can change the way you're staining. In this case, the way we fix it was adding more antibody that was still within the saturation. Another example, TCR gamma delta. In gray, only that antibody, good separation, negatives and positives. This is a critical one because you don't get a lot of positivity. And there is a dim population that might not be captured sufficiently if you don't use the good titer or a good staining protocol. When we go for fully or a multicolor sample, you see the spread in the negative in the black one increase and the positives went down. So increase the titer, change the fluor, or change the way you stain the cells. So we did sequential staining. I'm showing you, let's focus on donor A on the upper part. This is supposed to be my TCR gamma populations. This is CD3 for co-staining. I'm not seeing them if the antibody is added at the same time that all the others. Again, low expressor that cannot be detected well. But if I add the TCR gamma delta first, before adding others, I'll be able to capture that well. I see it here, clearly separating those two subsets of TCR gamma. So it's how you stain them, how much you add them, and not only the combination of fluors. If these strategies don't work, and you could include also strategies in sequential staining, like for chemokine receptors, changing the temperature at which markers are incubated. So all of that can help. But if not, then you might need to reassign fluors. This was the first time a 40-color panel was being published and out there. And it was a big hallmark, but we needed to make sure that the combination was really successful. So part of that is go and review. I told you about 421 and 436. Combination that is difficult to use, impossible to use in some instruments, and doable with careful panel design on others. So we assigned them to markers that were not co-expressed, but we needed to verify that any spread here and here was not affecting others, that we clearly can separate those populations. Important also to include FMOs is needed, because the presence of a particular fluor, like FITC in this case, could affect your ability to see others, like BV515. So if I exclude that fluor from the mix, do I see the same populations? Do I see a similar pattern? Or do I stop seeing something here just because this particular fluor was there? Same thing with other combinations. So important that you go for those combinations that are not that might not be that successful in terms of similarity, and check with FMOs if that's working or not. And FMOs are also critical for gating. So if I have a multicolor with all the fluors and an FMO that doesn't have the CXCR5, highly important for dimly expressed molecules, we see the expression of in CD4 positive cells of CXCR5, but the only way I can define where the positivity starts is by using an FMO. So that's part of the strategy of having a successful 40-color panel. And then at the end, what's the goal? It's after doing all that testing, finding perfect controls or optimal controls, finding the best combination between fluors and antigens, having a data set that, and it's out there if you want to take a look, but a data set that no matter what marker you look at, you get great separation for markers or molecules that are dimly expressed, or that are co-expressed, clear separation between what's single expressor and double or co-expressed, a good separation for populations that are not that frequent, and it might be more difficult to separate them, and the same across all the panel, right? If you're interested in NKs, in dendritic cells, in regulatory T cells, gamma deltas, for each of them, gamma deltas, we don't have many, at least in this particular sample. So very important that if I care about these 4 subsets, double negatives, double positives, and the singles, I can distinguish them correctly without having weird gate placement, just because the resolution in the instrument, in the selection of fluors, and in the particular arrangement of those fluors was affecting. So this is in general showing you things that you can see in the publication, but it's for every single plot. If you look across, you pick one plot, you will be able to see that things are separating, which is what we care, no matter the markers. We have different panels, but it's how critical it is to go through those steps. And especially the optimization, because it's what really tells you if good data is what you're getting. And just a little bit of marketing. You can get information from our website. Every single thing that I described, we have a video that matches, a 10-minute video, so you can learn more. It's an educational portal in our website. The Spectrum Viewer is great. You can create the theoretical panel there and have an idea if it's good or not. But definitely take a look at the panel builder. It's a great tool for building panels. It's free and it has everything in there: brightness, spread, and the emission profile. So it's gonna simplify your work. Remember, the rules are the same for 40 color or for 5. It's going to be easier for 5. You can spread things around easily. Make sure you follow the steps, know your system, your fluors, your markers, the rules, select well your controls, and remember that single stain cells are super critical. So that would be great to include. Yeah, thank you so much. Really? Okay.
So, all right. So first on from online question was, can we check the similarity index between fluorophores while picking for regular 13 to 15 color flow?
Yeah. The similarity index will be useful for anything, even if it's just 2. Or if it's 50, 55, like customers are doing now. It's a tool that allows you to compare between 2 fluors. So it will apply to only 2 or 10, any complexity. Yeah.
So please remember, anyone else wanting to ask questions, there are microphones down here on either side of the stage. All right, another question from online. Did you have trouble unmixing live dead blue from autofluorescent signatures of larger cell types?
That's a point that we didn't talk about, and it's panel design will also be influenced by how your cells look without any staining, so unstained cells. And it's gonna get trickier if you have, for example, alveolar macrophages that have a lot of yellow, green, and red emission, just unstained. So extra rules will apply, right? You don't want to use, you might want to reserve the red dyes for those cells. You might have a lot of emission out of the UV, UV7, UV8, violet 7, violet 8. So all of that will increase your background. And again, what we care about is the separation, right? Positives and negatives. So if the negatives are higher, then your ability to see the positives will decrease. Autofluorescence extraction will help, but it's still important. Certain considerations on panel design that depend on unstable cells. And yeah, for viability dyes, since we want to remove what's positive because it's the dead cell, so it might not have that much impact on spread. But we have seen that if it's not titrated properly, then it will impact how the data look, even if you're getting out the positives. Yeah.
All right, so another question from online is, what is the difference from stain index reduction spread matrix from spillover spread matrix?
It's the way in which it's calculated. For SSM, what you have is it's negatives and positives for a particular single color control. And the negatives have certain spread. The positives, let's say, are they still seeing my screen?
Yes.
So it's 2 different formulas, and the way they are calculated, it's because of that different in the information you need for that is different. So SSM, you have a negative and a positive. The negative has certain the spread in terms of RSD. What you want to calculate for the positives, it's how much the delta in spread. So as we see, the delta between this spread and this spread on the y-axis is different. So the SSM will be higher. The SSM was initially defined to compare between instruments. So it's going to be normalized by the intensity. So as Nick was saying, it's not influenced by intensity. So if you compare 2 fluors, one bright, one the highest, the number might be the same in terms of spread. But in reality, what you see in the plots is different That's why we use a different metric. So SR, the stain index reduction. If you compare, let's say for APC, you have negatives and positives, single color scenario, so nothing affecting them. What you could do to see the separation is the stain index, right? The positives and negatives divided by the spread of the negative. The SRI, it's how much that stain index in the single color scenario, it's reduced by the presence of another fluid. So the way it's calculated is you take the difference between the positives and the negatives, similar to stain index, but you don't divide by this one. You divide by the spread of the positive. So once you do that, you take into account, so for this one, you need 2 fluors, not only one. You take into account the spread of the population positive for another fluor. Then we calculate the percentage. So we will tell you the SIR, the stain index reduction, will tell you how much the stain index was reduced in the presence of other fluor. So it's 2 different comparisons. This one depends on brightness. So that's kind of the difference between them.
We have a couple more questions, but we may have to limit this to.
Isotopes. Oh.
All right, let's see here. Do you have a favorite one you want a quick answer before? It's no one else. You get to pick one. Speaker choice.
How do you determine the 7?
7?
Titration?
All right. So a final question for Diana. In a titration experiment, if you do not see saturation, how do you determine the ideal antibody concentration? Do you take the highest concentration or volume? Question mark.
Very, very good point. Couple of things to do, and depends on the marker. So, for example, if you're titrating HLA-DR, if you go and try to increase the concentration, that's gonna saturate the thing. It's gonna be way out of saturation. And sometimes when we don't see that saturation phase, is either because you are not testing enough or adding enough antibody concentration, or just because of the antibody characteristics, right? It's a low binding. The ability to bind is not that as constant. So it's dissociating, and then you never reach that saturation. Also, it's going to depend on whether you have IgM, IgG. IgM will give you brighter. And then, but back to the question, what I've seen for highly expressed markers or even secondary, like HLA-DR, tends to be the case because it's way too bright. Going higher is not going to change, not going to add anything. So we pick the titer based on markers that are co-expressed, knowing that we are not at a saturation point. Then at least pick a titer that it's not affecting others that much. So going lower, 10 to the 5th. But for markers like TCR gamma delta, you never get there because you went up to, usually the most of the antibodies can be titrated like 1,000 nanograms per test, 1 milligram. But even if you go, so you might go higher, 2,000. For TCR gamma delta, it's not going to matter in terms of getting out of scale because you'll not get that high expression level. So going higher is one option, but also changing to maybe another clone. It's an option that keeps you, it tends to be the case for CD20. And that was published years ago that 20 never reaches saturation for certain clones. So using another clone is an option. But yeah, multiple things that can be done. And when you don't get the saturation going higher, finding a better clone or another clone, or just making the decision based on what other markers you have in the panel. Yeah.
Okay. All right. For the last speaker of this session, prior to, we're going to try to get set up. All right. So, well, that was a great talk. All right. So, for next, we're going to have a short talk from Omar Abouzid. Hope I pronounced that right. On Easy Panel. This is a product that's out there for helping design panels that integrates not only fluorescent intensities but also antigen densities. And personally, for my core facility, it's something that we've invested in and are going to go with as in just another one of those tools for high parameter design. Omar is the co-founder of Easy Panel, a Paris-based startup developing a fully automated intelligent tool for flow. It's now licensed to 70 cores. Sorry, trying to get, so anyways, the, Omar is an MBA candidate at MIT and a master's graduate from the bioscience in INSA Lyon, France. And Omar, please. There you go.
All right. Good morning, everyone. I hate this spot because it's the one standing between you guys and lunch. So I'm gonna try to be concise and keep it short and sweet. So thanks so much for the introduction, Joel. So yeah, I'm gonna be presenting EasyPanel, which is an automated and intelligent tool that we co-founded about a couple years ago. That uses artificial intelligence and automation to suggest panels from 2 colors, which is easy, to 52. This is the highest that we've seen. So I think I'm going to break it down very quickly here into what is the problem that we're trying to solve, what is EasyPanel, a quick demo, and then a couple test cases, and then take questions. So the problem that we've seen, my background, as Joel said, is in immunology and flow cytometry. And the problem that we've seen is that most or a lot of panel designs, at least a couple of years ago, they were not automated and they did not necessarily lead to quality panels, meaning that you needed to have some level of experience. You needed to select the fluorochromes yourself. You needed to know what is the difference between PEC F594 and PEC CY5. And so they mostly resembled commercial catalogs rather than intelligent tools. And you guys all know ChatGPT and, you know, the self-driving cars of Tesla. So the world is really advancing. And I was finding myself personally spending 6 hours a day designing panels and choosing fluorochromes. And then my PI scolding me because I was putting 2 fluorochromes that were not really compatible. So I was thinking, what is, you know, maybe we can come up with a smarter tool that could maybe figure out the optimized combination based on a few different criteria and suggest a panel ideally in a short period of time. And ideally also in a way that could be, you know, vendor and fluorochromes agnostic. So, you know, without specifying tools here, we thought that the 2 criteria over which we could rank panel designs are automation or intelligence, which is the axis that you see here. And ideally, we'd want to see things that are highly automated that could take as the least time possible, and also quality without restricting ourselves to one vendor or one company versus another. Because, you know, all companies make good fluorochromes, so we'd want to, you know, broaden our options as much as possible. And, you know, if you guys go online, you'll see on Reddit and ResearchGate, and now these are outdated, but you'll see like hundreds, if not, you know, more of people really asking about like automation in panel design. And so what is EasyPanel? It's, again, an intelligent and fully automated algorithm or tool for flow cytometry panel design that we launched a couple of years ago. We've gathered quite some data now as we work with maybe 70 or more flow core facilities and companies. And so, you know, as we gain data, we get smarter, hopefully. And so, again, it's fully automated. It's vendor and fluorochromes agnostic. It integrates with OMIPS and the latest flow cytometry publications. And I want to also mention that it's fully automated, but you can also manually edit it. And I'll get back to this very important point, because panel design can be a very religious topic from my experience, at least for the academics. All respect due to them, of course. It also adapts to your own experimentally generated SSM and data. And so with this, I thought, you know, enough blah, blah. I can just go through the actual thing, which is just right here. So the tool is posted at flow-cytometry.net. just a French keyboard. All right, there you go. So yeah, this is the homepage. Flow. Let's see if I can. Oh, didn't get that right. This is the web page. It's accessible to everyone. There is a limited free trial version of it where you can see a few different fluorochromes. You can read here. You can also access a few short video tutorials on the resources page. But then once you log in, if your company or university has a license, you'll find your page where you'd have the list of your different flow cytometers here. So I'm referring to Joel's Flowcore because we work with them, as he mentioned, among other universities in Texas. So the first thing is that you'd select your flow cytometer. They happen to have an Aurora, but also a few different cytometers. So maybe I can go through the Aurora first and then maybe check a traditional cytometer. So once you select that, shows you the configuration, and then you have a couple different options. One is minimizing or optimizing the panel based on the similarity and the complexity, which are published data and they are on Cytec's website. And again, this is a one-stop shop for all cytometers. This is not, again, specific to Cytec. This is also works for BD, thermal, whatever cytometers you may be using, as you see here. I'm just taking this as an example. So again, the first option, which is the default, consists of minimizing similarity and complexity, as was shown by nicely shown by Diana a few minutes ago. But then you can also upload your own experimentally generated SSM or spillover spread matrix, because as you all may know, spread can be a factor of laser intensities and instrument configuration, especially for traditional cytometers. As you ramp up PMTs or decrease laser intensities, really you can get spread that is very variable, even for the same brand, the same instrument, from one core to another. And so for this, we made this tab called Custom or Experimental SSM Manager available. Not all flow cores have this. I'd say maybe less than 15% from the ones we work with. But in case you do have the data, you can go on to this tab, usually make it accessible to flow core managers. And you'd have to use this template. It's a very intuitive template. It's an N-by-N matrix listing the fluorochromes that you ran on your cytometer. And these would be your spread values that you assessed or measured yourself. Not only this, but there's also the stain index. And again, the stain index can be a variant or a factor of your laser intensity. So if you're able to calculate this on your own cytometer, on your own machine, you can put the data here and upload the Excel. EasyPanel will take these values into account for your panel design. For now, I'll just go with the default. I just wanted to mention that this is possible in case you have the data. Page number 2 is where you add the antigens. So again, we tried to make this as intuitive as possible. So you can add, there are 3 different ways of adding the antigens. You can add them one by one, just like I'm doing here. It recognizes all the aliases. You can even enter this as a free text. We noticed that some people are working with MHC tetramers or like some proprietary antigens. So you can call this, I don't know, FlowTex T4 lunch antigen. And you can enter that. Of course, there will not be commercial availability for this because, as far as I know, BioLegend doesn't sell this product. But it's just to say, you can even enter autofluorescence, because there was a question on that a couple minutes ago. And what we've seen people do is that they assign a specific color to autofluorescence. For example, here under specify fluorochrome, this is an optional field, but in case you think your autofluorescence comes from BV 650 or 605, usually we've seen a lot coming in the violet. You can select this, and in this case, the tool will take into account the fact that you have some autofluorescence from your cells in having a spectrum similar to the BV or whatever that color may be. The second mode is importing in Excel. We've seen 40 color panels a couple of minutes ago. So in case you have this, you can upload this via an Excel. We'll populate the whole page. Another input mode that we included recently, actually based on feedback that we've gathered, is selecting cell populations. This works nicely for people who are more on the exploratory side or who may not be really sure what they're looking for. So clicking this, you'd get an option for a human or mouse. Say human, you would be asked for the cell population. Say you're working on, I don't know, you have all the options here: NK, dendritic, macrophages, monocytes, whatnot. Say T helper cells, you get asked for the subset. You have an option for default or general, but you can also specify I'm working on TH1, for example. Then you get a listing of all the cell surface markers that, since we have access to a lot of online databases and literature, we try to list them all here, and we have even someone dedicated to monitoring the literature. And as soon as a big paper comes out identifying one big marker for cell subset, it gets updated automatically here. You can also add or not the secreted proteins of this specific subset and then the transcription factors. I'll just go with the cell surface markers right now. I added all of them here. They got all added. You can delete some. You can add some, just like I'm doing here. And you can, have your panel all set. You can specify, so as some of you may have noticed here, the expression level was added by default for a few antigens. So again, the algorithm is intelligent in the fact that it has access to a lot of databases and publications. And it turns out that in a lot of publications and online databases, CD3 was marked as highly expressed. Surprise, surprise. In case that's not the case in your experiment, you can always reset it or say unspecified. CD3 may happen to be downregulated in case T cells are activated. It's just that in most cases, people refer to CD3 as highly expressed. And so we enter this by default for 70 or 80 different antigens. So CD3 was high, CD4 was high, CD8 was high. I'm going to delete this FlowTex before lunch. This guy was low, CXCR3. And again, if you don't agree on these choices, you can always reset it to unspecified or, just say that I don't know. We just try to help as much as possible. You can reset the species reactivity to mouse, rat, or whatever species you're interested in. You can set different antigens as a dump channel, just like I'm doing. And for those who don't know what a dump channel is, there is a small question mark right next to each field to explain what this is, because it turns out that this tool happens to be useful not only for people who are designing large panels, but we saw that, for our surprise, a lot of people designing 3-4 color panels actually use that because they don't really have experience in flow. And I just wanted to say that right next to each single field, there is a small question mark explaining what this field is and how exactly it impacts the panel design. You can mark different antigens as co-expressed on the same cell population called group 1, and then group 2, a second lineage, and then group 3. Now, you don't have to do this. This is more really for fine-tuning. If you really want to tell EasyPanel, be careful, these 2 or 3 antigens are co-expressed, really separate them for me as much as possible. This is something that you can do. By default, we saw that the algorithm is geared towards minimizing systematically complexity, similarity, spillover, depending on what cytometer you're working on. So it really tries to choose fluorochromes that are distinct as much as possible. So I'm going to unresolve that. And then you can specify fluorochromes. So one important thing, you don't have to be using this tool for panel design from scratch. Happens a lot that different universities and different companies, they already have a stock of antibodies, right? They don't want to buy things from scratch. So they can say, oh, I actually happen to have CD3 FITC in my fridge, but I also have this with APC, and then I have this with whatever they make, that may be C-Fluor B515. Oh, the tool is not happy with that because it tells you that there's a lot of similarity between this fluorochrome that I chose randomly in FITC, but I'm just going to choose something else here. All right, I'm going to choose this and maybe get like APC, whatever, size 7. And then let's add a NovaFluor so that our friends at Thermo are happy. And so I chose a few fluorochromes at different random places, and I left the others unspecified. And so in this case, this is what we call a partial panel design, meaning that you put what you already have in your fridge and you're asking the tool to design the others in the best possible way. A third scenario that we've seen people do is putting your fully designed panel and not asking EasyPanel to design anything for you. In this case, it will give you the quality score of your panel, and it will suggest what it thinks to be the best possible panel compared to what you already have. All right, let's move forward. Page 3 out of 4, we're almost there. Viability die. So same logic as before. And by the way, here before, you can really choose from a huge database of different fluorochromes, including, viability, fluorescent proteins, and, all the relatively new fluorochromes from the different vendors. Viability die is the same thing. You can either ask EasyPanel to determine, optimize one for you. You can specify your own, or you can say, I actually don't need the viability die. You can also exclude fluorochromes. So say, again, we're fluorochromes agnostic, but it turns out that some people are not comfortable working with specific fluorochromes for whatever that reason may be. I'm just gonna do a random selection here. So you can really exclude any fluorochromes that you don't want to see. And then again, all respect due to all vendors. This was a request by quite some people. You may exclude vendors specifically, in case, yeah. Again, we're vendor agnostic, but this just turns out to be helpful for some people. So I'm not gonna exclude anyone. I don't want to get myself into trouble here, but this is an option. And so you move forward, and this is where the automation happens. Takes 30 seconds at most, depends on how much the servers are being consumed at that point in time. We should have run a small short music or commercials or something. All right, so usually takes less than 40 seconds maximum. That's the longest that we've seen it take. We try to parallelize the servers. And so you end up with a table that is as easy to follow as possible. It's color-coded with the different colors of your lasers, in this case, the Aurora 4 lasers that Baylor has. The list of antigens, the list of fluorochromes, and then the commercial product suggestions. So just let me give you an example here, just CD3FTSE. We'll give you also, there's a small icon next to the different fluorochromes showing you the estimated stain index of this fluorochrome. The star sign is here just right next to FTSE, because if you guys remember, I'm the one who fixed FTSE in this particular position. So I just placed a few fluorochromes in some positions. So here, CD3FTSE, what happens if you click? You'll see the commercial product suggestions for this. So not only do you get the panel, but also the commercial product suggestions of where exactly you can buy them. We also go one step ahead by even suggesting things. So this, in this window, you get the information of the vendor, the catalog number, the clone, the isotype, the size, and then whether or not this specific clone has been listed in an OMIP or not. And we have a database of all the OMIPs, and we're expanding this database even into more and more papers. So it just happens that this specific clone of CD3 human has been cited into OMIP 68. If you click, you get redirected to the specific paper. And you can find all the details here, including the clone. You even get, we're updating the list prices. They just change and sometimes they vary by location, so this is tricky. You also get the link to the particular product on the vendor's website so that you can purchase it directly. We also have some reviews that were uploaded by some of our users that you can view them here in case they are available. Last, I want to say there's the complexity index of this panel. It's 6.73. This is a relevant metric, as Diana said, for the SciTech. We have other metrics for BD or for the panel if it was fully designed by EasyPanel, meaning that if you guys remember, I fixed some fluorochromes in specific positions, and I've done this randomly just for the sake of the demo. Easy Panel is trying to tell you here, hey, if you would have let us just run the full panel by ourselves, just hit this button, view, we will show you that we would have been able maybe to suggest a better panel. And in this case, it would have been a panel of 4.5 complexity compared to our partially designed panel of 6.7. And it will get opened here in less than 20 seconds. Similarity matrix, it's shown here and it's color coded with a heat map. You can download the full Excel and you can get it integrated into your notebook. It includes your cytometer, the complexity index, the similarity, the different antigens we listed, the fluorochromes, and not only that, but also the commercial product suggestions with the clone and with the specific link to each vendor. You can obviously download the similarity matrix. You get access to the spectral profile. And again, this is specific to the Aurora. If we would have done any other instrument, would have been relevant to that particular instrument. This is the panel that would have been designed by Easy Panel. It has a lower complexity index, as you would have seen. We have also automated gating suggestions. So the tool is intelligent in that it recognizes which populations you may be interested in. And if it finds enough similarities with a paper that has been published or a figure that is in an online database, it will give you this image or this that you see here. For example, here looks like it thought that we're interested in CD4 T cell subsets and then NK cells. So it shows you this. This is a bonus point that we show you. We tell you, hey, you may get data that looks like this. Finally, you get also papers matching your panel design, OMIP, and we have a database of, I think, 500 papers now. We don't dump these into the face of users because otherwise they don't never read them. I would have never read, like if someone was suggesting 85 papers to me, would have not opened anyone. But these are just the papers that are relevant to you. And what I mean by relevant is that you need to be sharing at least 10 or more antigens for us to suggest a paper. Because we would be saying, hey, your panel is similar to something that has been published already, and your antigens are almost the same. So you have the link here. You can click on it, refer to the paper. One last thing that I mentioned, from our experience, panel design can be a religious topic. Some people can say, oh, I would have never matched CCR1 here. CCR1 should have definitely been with PE. Seen that a lot. We think this panel is great. We think that it saves people a lot of time, takes maximum 30 seconds. Now, it's very hard to emulate human experience, no matter what tool you're using, ChatGPT, Easy Panel, whatever that is. So we made this button accessible for everyone so that it's called Customized by Manual Panel Builder. Once you get this, click this button. It will open the panel that we designed for you, and it will show you the full commercial catalog of everything that exists. And it will tell you, hey, go ahead, optimize or edit as you see fit, up to you based on your own opinions and experience. So this will get loaded. Say you don't like CD4 PRCP, you think it's a crappy choice here. We say that's fine, just remove it, just like I done. And say you prefer CD4 BB515. We show you the different products. You prefer this. We'll give you warnings in case you make like not so great selections. We thought about putting like an unhappy face here, but again, it's up to you. You're the one who drives this. I'll just like make a couple random selections in CD4 BB5. I don't like this. I'll put it here. And then you can move next. And what we'll do is that we'll compute the corresponding complexity index once you have edited yourself the panel, corresponding spectrum. And in this way, you'd be able to compare with what you initially. So yeah, this is pretty much it. And yeah, we have pretty interesting case studies that we're trying to push for publication. Unfortunately, one of them is with a big pharma company, so they're a bit reluctant, but they had really, really interesting results comparing a panel that has been published, happens to be in an OMIP, versus a panel that was designed using Easy Panel. Actually, to be totally honest, a combination of fully automated Easy Panel and manual panel design happens to give the best possible results. So, in this case, it was OMIP 69, which is a great panel, complexity index 54. Here it had, actually now with the newer version, it had 15. This took quite a few hours. I put a star because I, didn't really ask the authors how much time or if they, with a stopwatch on their panel design, but took 38 seconds in the case of Easy Panel for a 40 color panel. They've done some manual panel design. They told me they took 20 minutes to do that, mixing and matching and fine tuning. And the results, this is a quotation. I was not able to cite the exact name of the person. Again, we're pushing for publication, but it led to significantly better signal and much, much better cell resolution and cell populations resolution in a much lower time. So yeah, this is great. Thanks so much.
I'm going to have to ask the forgo questions. Omar will be around, as well as, sorry, we will ask him for some of the online participants as well. We will get some questions answered, but we are late for lunch. So in that, please, everyone, we will push back the start time by 5 minutes of our next one. But we, as of right now, I believe we have a half an hour now for lunch. Thank you. You added some, you have tweaked it. It's looking so much better.
Yeah, we have some exciting things even coming up.
Yeah,
So
awesome. Again, I'm hoping I can get my people to start using it and utilizing it.
Oh,
Just you guys actually see each other.
yeah, we staged quite a few.
Yeah, we did. We did. I mean, we still appreciate I did not realize we did the getting strategies and stuff. That was like new.
we actually have quite a few things even, but I didn't have time. I saw
That's
that.
all right. We're gonna, no matter what, we're gonna have a web.
Oh, yeah.
Yeah.
Hey, Leroy.
Today was cloudy and cold and it wasn't